స్టార్ట్ చేయాలా? లే ఏంది ఎవరో గుర్తున్నారు. లేచేయమంటారా? ఏమైంది?
Dear students, for the prayer song. It is now my honor to invite our distinguished speaker, Dr. Krishna Chaitanya Karli, Associate Professor, Department of Chemistry, College of Natural and Computational Sciences, Oxfam University, Oxfam, Tigray, Ethiopia, to deliver the invited talk virtually. Sir, may I kindly request you to join us on the virtual platform. Thank you, sir. It is now my privilege to invite Dr. Vani Lata, Head Department of Chemistry, to the dais as the co-chairperson for this session. I invite B. Kokila of Microbiology to welcome Madam with a sapling. Thank you, Madam. Thank you, Kokila. It is now my honor to invite our co-chairperson, Dr. Vani Lata, to introduce our distinguished invited speaker, Dr. Krishna Chaitanya.
Pleasant good morning to one and all.
It's my privilege to introduce our esteemed resource person, Dr. Krishna Chaitanya Kari, who brings over 15 years of teaching and research experience in the field of biochemistry. He currently serves as an associate professor and researcher in biochemistry and organic chemistry in the Department of Chemistry at some university, Ethiopia. Dr. Kari's research is dedicated to the development of novel immune checkpoint blockers, immune tolerogens, anti-cancer and anti-inflammatory nanoparticles, and antimicrobial natural and synthetic agents. His work primarily focuses on controlling and treating cancer, autoimmune inflammatory disorders, and infectious diseases. Notably, he has contributed to enhancing the efficacy of cancer immune checkpoint inhibitors in female reproductive and breast cancers by targeting the tumor microenvironment. His academic contributions extend beyond research, with active collaborations with prestigious institutions such as University of Kiel, Germany, PGIMER, Chandigarh, India, Geetham University, and Central University of Kerala. Dr. Kari is also an active participant in academic discussions and skill enhancement programs, having attended 16 workshops and 20 conferences or seminars. His scholarly work includes 20 national and international publications and an extensive list of 80 research articles. His impactful contributions to the scientific community continue to inspire researchers and students alike. We are proud and honored to have Dr. Krishna Chaitanya Kari with us today. Please join me in extending warm welcome to. Now I invite Dr. Krishna Chaitanya Kari to share his valuable insights and deliver his presentation.
हाँ जी।
issue over there is written like is unstable. So we are calling forward for online and on-stage oral presentations. So I now invite Ms. Mini Fernandez to carry on with her oral and paper presentation. Good morning, everyone. Please, can you confirm if you can see the screen? Hello? Can you please confirm if you all can see my screen and hear me as well? Hello? 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 Rekha ma'am, can you please let me know if I can start my presentation? Hello? Hello? Can you please let me know if I can start my presentation? Hello? Hello? Hello?
Hello. Yes, ma'am, it's audible.
Yes, ma'am. Can I begin with my presentation?
Yes, ma'am.
Okay, thank you so much. Good morning, everyone. I am going to give a presentation on investigation of plant defensin, fungal cell wall receptor interactions, and the role of these plant defensins in imparting pathogenic resistance. My name is Mini Fernandez, and I'm a research scholar at the Department of Life Sciences Biotechnology Division at Geetham School of Sciences, Vishakhapatnam. So to start off with, we'll have a brief introduction of what plant defensins are. So, plant defensins are cysteine-rich cationic peptides act, which act as a key part of plant's innate immune system, offering antifungal and antibacterial protection. So, just like how we humans have our own innate immune system, which fights against various pathogenic infections, plants also have their own innate immune system, and one component that contributes to these plant innate immune system are these plant defenses. And these plant defenses, they are cysteine-rich cationic peptides. Now, the structure of the plant defensins, as you can see on your screen, these are just few of the plant defensins which have been there in the literature. These plant defensins, they are small cationic proteins made up of roughly around 45 to 54 amino acid residues, and these amino acid residues in their structure, as you can see, which is depicted by yellow color bonds, they are held together by disulfide bonds. And the presence of these disulfide bonds make them highly stable against various proteases and extreme pH and temperature conditions. Now, where are these plant defensins localizedly present in the plant? So they are found anywhere in the plant. They are most commonly found in seeds, and they are also found in other plant tissues such as leaves, roots, bark, pods, tubers, fruit, and also floral organs. Now, coming to how these plant defensins actually play a role in imparting defense or immunity to the plant. So, these plant defensins can show a broad range of antimicrobial activity through multiple mechanisms. And out of those multiple mechanisms, one mechanism that most of the plant defensins target is through cell wall. So the plant defenses interact with the fungal cell wall, and they enter inside the microbial pathogen, and then by the ROS treatment or by apoptosis, they cause fungal cell death.
Now, why are we learning about plant defenses? Now, the idea of learning about plant defenses and their mode of action in detail had come up because of the devastating Panama disease that we can see among banana plantation. Now, this Panama disease is also called as fusarium wilt, which is caused by the fungal pathogen Fusarium oxysporium. Now, the spread of this Panama disease is so vast among the banana cultivators that it has caused a pandemic in the banana cultivation. And so that is why there is an urgent need of developing strategies that can be employed to curb the action of the spread of the
Excuse me, ma'am.
fungal. Yes, sir. Yes, sir.
Now it's okay. You can proceed, ma'am.
I can continue, ma'am.
Yes,
Yeah, yeah, okay, ma'am. Here with
Okay.
you.
Okay. So, because of the devastating fusarium wilt that we can see among the banana cultivators, we have intensively studied the role of how we can use these plant defenses in order to make sure that the banana plant can provide resistance to itself instead of using fertilizers or harmful pesticides, which can further cause soil infection. Now, as I told you, why do we have to clone defensins? Because it has so many advantages that is we can develop disease resistant plants. It provides innate immunity for the plants. It regulates plant growth and development, and it can be toxic to the invading pathogens. And if you see, the role of defensins in crop improvement has not been extensively studied until now. So hence we have taken up the study of understanding how we can use these plant defenses in order to develop a transgenic plant which can be able to fight against pathogen infection. So the different methodology that we have used in our study is we have first done in silico studies where we have understood the interaction between the different plant defensin genes and the fungal cell wall receptors. So the two fungal cell wall receptors which are common in most of the fungal species or pathogens is the MAPK receptor and the phosphatidic acid receptor. So we have targeted these two receptors and we have studied the interaction of these plant defensins to these fungal cell wall receptors. So after in silico studies, we have performed many in vitro studies where we have done, first of all, the isolation and characterization of genomic DNA. So the plant defensin that we have used is MS-DEF1 plant defensin gene. Now this MS-DEF1 plant defensin is isolated from a plant, Medicago sativa. So the seeds of Medicago sativa was obtained and from the seeds we have isolated the genomic DNA, and after this a gradient PCR of the defensin gene was performed. After this, so in the gradient PCR we analyzed the optimum annealing temperature for the amplification of the defensin gene. Followed by done, we had done a RT-PCR analysis for which we have isolated the RNA from the seed again, and then we have continued with DNase treatment, and then we have performed QRT-PCR. So once we have got an amplified product, then we have cloned the defensin gene into a pCambia vector. And after the cloning was successful, we have transformed the recombinant pCambia vector into our host cells, which is E. coli. After transformation, we have successfully transfected the transformed E. coli cells first into Agrobacterium and then into the model plant of our study, which is Arabidopsis. So coming to the results. So first is the in silico interaction that we had studied between the plant defensins and the fungal cell wall receptors. So on the left side of the screen, we can see there are the different 3D structures of plant defensin genes. So we have studied around 12 plant defensin genes. So we have first we have retrieved the protein sequences of these plant defensin genes from various biological databases. And from there, we have generated models for these plant defensive genes by using Swiss model database. After which, these models were docked with the fungal cell wall receptors. As I told you, we had studied two fungal cell wall receptors. One is MAPK, that is mitogen activator protein kinase, and phosphatidic acid receptor. So these are the two receptors that we had targeted. And then we had, so once the docking was performed, we have seen that um, so for docking, the software that we have used is Autodoc Vina. So in Autodoc Vina, we have seen that uh, the docked molecule showed um, low binding affinity and zero RMSD value. So a negative binding affinity indicates that there are stable interactions between the protein and the cell wall receptor. And a zero RMSD value also indicates that the dock has been done perfectly. So after the docking was done, the interactions were viewed in Discovery Studio, where we had seen that all the plant defensin proteins were showing interaction with the fungal cell wall receptors, and the bonds that have been found which are interacting between these two were mostly hydrogen bonds and alkyl bonds. So after we had finished the in silico analysis, the next step was in vitro analysis, where we had isolation and characterization of the genomic DNA. So the gel picture here shows the DNA which has been isolated from the germinated seeds of Medicago plant. So lane 1 denotes the ladder, and lane 2 and 3 shows the appearance of DNA bands. So followed by isolation and characterization, we did a gradient PCR. So this is the primer sequence that we had used for amplification of the DEF1 gene present in Medicago sativa plant. And below is the PCR condition tables which we had used. And we had used a gradient of annealing temperatures from 55 to 59 degrees. And after the amplification was done, an agarose gel electrophoresis was performed, where we had seen that we had got a good amplification, good amount of amplification at 59 degrees Celsius. Followed by gradient PCR, we next used QRT-PCR, where we had used defensin as our gene and actin as our internal control. And these are the PCR conditions for RT-PCR. So once RT-PCR was done, an amplification pot was obtained. So once the amplification pot was obtained, we had calculated the CT values and the delta CT values and all the fold variation, all of that was calculated. So the delta CT value is calculated by the difference of the CT value of our desired gene with the CT value of the internal control, which is actin. And then we calculate the fold change by using the formula 2 power minus delta delta CT. So once it was calculated, we had found that the CT values of actin was found to be around 0.1395. For my defensin gene was minus 3.751, and their expression was around 14.83, and the fold variation was 1.17. So these values have been showed in the amplification plot and also in the bar diagrams that have been shown. The melt curve analysis was also performed. So in the melt curve analysis, a single peak was obtained, which shows that there was no uh, enormous amplification of the gene. Followed by QRT-PCR, we have now done the cloning experiment, where we have cloned my desired MS-DEF1 gene into pCambia vector. So we had obtained the pCambia vector, and from there we have used restriction enzymes to digest um, the vector, the multiple cloning site of the vector. And after the digestion was done, we had inserted the MS-DEF1 gene, or we have ligated the MS-DEF1 gene into the vector. Once the ligation was successful, then we had transformed this recombinant vector into the host cell, which is E. coli. So after we had cloned, now if you can see, the pCambia vector has a resistant marker of kanamycin. So kanamycin here acts as a selectable marker. So once the recombinant vector was performed, was obtained, and it was transformed into E. coli cells, we had plated them on a selectable media. So as you can see on the left side is a control where no colonies were seen because of the presence of kanamycin, because we know that our host cells, which is E. coli, do not grow on an antibiotic medium because they are not resistant to it. Whereas we had seen the formation of colonies on kanamycin uh, media added to LB media. So these colonies indicate the presence of transformed cells. So after this was done, after we had obtained these results, then the next step was transfection of this recombinant vector into Arabidopsis plant with Agrobacterium tumefaciens. So before we had done the transfection, first we had transform this host cell into or the desired gene into Agrobacterium tumefaciens. For that, we have used infiltration media. So to this infiltration media, we had added one colony of the transformed bacteria and we had kept it in shaker overnight. So by co-culturing method, we have seen that the there is a transfer of the vector into the Agrobacterium tumefaciens. So once that was done, the recombinant Agrobacterium tumefaciens was now transfected into the Arabidopsis plant. So the plant leaves of Arabidopsis were dipped in the bacterial suspension for around 30 seconds, and and then it was grown overnight in media containing kanamycin. So if you can see on the left side of the picture, this is a transfection of P. cambia vector into Arabidopsis plant. And the right side, we can see that a selection of transfected plant was done from a non-transfected plant. So a transfected plant successfully grew in the MS media containing kanamycin because it has the vector
Hello,
coding
Vicky.
for. Yes, ma'am.
Can you come to the conclusion? It is taking
Oh,
long.
oh, I'm so sorry, ma'am. Yes. So,
Thank you, ma'am. Take it.
so finally, I conclude that this this study can be used as a model plant. That is, Arabidopsis can be used as a model plant to check whether the transformed gene, that is defensin, is resistant to the bacterial pathogen. So further on, studies has to be done on where we are injecting or the fungal pathogen onto these transformed Arabidopsis plant to check for their activity, to check for their antifungal activity. That's it, ma'am. Thank you. Thank you so much.
Thank you, Ms. Devi. It is now my honor to invite our esteemed speaker, Dr. J. Navina Lavanya Lata, Principal, Government Degree College, Nuzweed, Andhra Pradesh. Madam, may I request you to come on to the dais? Sheikh Tabassum of Biotechnology will welcome Madam with a sapling. Thank you, Madam. Thank you, Tabassum. It is now my privilege to invite Dr. Sister Japamale, Head Department of Zoology, to the dais as the co-chairperson for this session. Praise of Microbiology will welcome Sister with a sapling. Thank you, Sister. Thank you, Praise. It is now my honor to invite our co-chairperson, Dr. Sister Japamale, to introduce our esteemed invited speaker, Dr. J. Navina Lavanya.
Good morning to each one of you. I'm happy to stand before you all to introduce the eminent personality, Dr. J. Navina Lavanya Lata. Dr. J. Navina Lavanya is an esteemed associate professor in the Department of Biotechnology at Krishna University, Machilipatnam, India. With over 15 years of academic and research experience, she has made remarkable contributions in the fields of biotechnology, environmental sciences, and molecular biology. Dr. Lavanya Lata holds a PhD in biochemistry from Osmania University and has previously worked as a scientist at Dr. Reddy's Laboratories and a postdoctoral fellow at CCMB Hyderabad. Her research focuses on metal-ion interactions, antimicrobial studies, bioremediation, and molecular docking. She has published 65 research papers, authored 5 books, and holds 8 patents in biotechnology and medical innovations. Recognized for her excellence, she has received several prestigious awards, including the NESA Green Technology Innovative Award in 2023, Excellence in Academic and Research in Biotechnology Award 2023, and Dr. B. Ramalingesh Swamy Memorial Award in 2019. Beyond her research, Dr. Lavanya Lata plays key leadership roles, serving as in charge principal of Krishna University, Dr. MAR College of PG Studies, Director of Admissions, Chair of multiple boards of studies, and IPSC coordinator at Krishna University. I'm happy to inform she is also the university nominee for our board of studies in geology. She is also actively involved in national and international conferences, serving on editorial boards of reputed journals. With her vast expertise and dedication to scientific advancements, Dr. Lavanya Lata continues to inspire and contribute significantly to the fields of biotechnology and life sciences. Dear Ma'am, we are happy to have you this morning with us and appreciate your willingness to come over here to share your expertise with us. We are eagerly waiting to hear from you. Please, ma'am.
Good morning, all of you. I thank the organizers, especially Rekha ma'am, for inviting me for this international conference, and also I thank Sister Japare for introducing me in a beautiful way. Coming to my topic, understanding the fungal infections, classification, diagnosis, and management. So, I use the bilingual language, okay? So, every day మనం చాలా చదువుతూ ఉంటాం, like bacterial infections and honey, viral infections and honey. Recently, we come across with the COVID-19. Okay, it is too dangerous. So, మనందరం ఇచ్చేది ఏంటంటే we give importance to the bacterial infections or viral infections. We sometimes ignore the fungal infections because we feel that they are not that much threatened. Okay, so but fungi are the omnivores. Bacteria and virus can survive only at 37 degrees or in the specific culture medium. But whereas the fungi, they can survive anywhere. సో మన ఇంట్లో బూజులో గొడవల మీద బూజులో చూస్తుంటాము, లేకపోతే ఫుడ్ మెటీరియల్స్ మీద చూస్తుంటాము, ఎనీవే తెలియదు. ఓకే, సో అండ్ కమింగ్ టు ద present scenario, the fungal infections are very important to study.
The study of fungal infections and their diagnosis is called the medical mycology. The medical mycology is a branch of science that deals with the study of medicinally important fungi. So, as we all know that fungus, the name derived from the mycus. Mycus means it's a mushroom. Fungi differ from the bacteria and virus because bacteria and virus belongs to prokaryotes, whereas the fungi are the eukaryotes. So they also possess all the eukaryotic organelle as similar with humans and all, and they possess a rigid cell wall. They contain chitin and beta glucans and others. And one more thing, they contain aldosterone, which is not the cholesterol. So, why the study of fungal infections is crucial when compared to the bacteria and other infections? Because bacterial infections have been extensively studied. So, there are so many types of bacteria, Acinetobacter, Staphylococcus, or Pseudomonas, or Klebsiella, many types. But they are well studied, and there are so many culture identification techniques or diagnostic techniques for the bacterial infections. Whereas the fungal infections are increasingly becoming alarming because their resistance to the survival conditions and very less treatment options are there and high mortality rates. So the main reasons are first one is rising global burden and mortality because the fungal infections affect over 1 billion people across the world annually and more than 150 million deaths are reported because of the serial fungal disease. And the mortality rate of invasive fungal infections ranges from the 1.6 million annually comparable to that of TB and malaria. Okay. Next, the immunocompromised patients like the HIV patients or cancer patients or organ transplantation affected patients, the fungal infections pose a greater risk to the death. The second one is limited treatment options because when bacterial infection can be viral infection can be done around and we used to take antibiotics like penicillin and or dose 65 paracetamol like that. There are so many options are available. But the fungal infections only three types: pollens, azoles, or echinococcus are available. And fungal infections develop resistance rapidly because as you see that మనం ఇంట్లో నాకు బ్రెడ్ అట్లా పడేసిన కానీ లేకపోతే కర్డ్ యూజ్ చేయకుండా ఇంట్లో చాలా ఫ్రిడ్జ్లో ఉంచినా సరే ఇట్ డెవలప్స్ ది ఫంగస్ సమ్ బ్లూ కలర్ ప్యాచెస్ ఆర్ yellow color patches will develop. So all are fungi only. And unlike the bacteria, fungi share similarities with that of the human cells, making it too difficult to develop the antifungal agents. And the third reason is emerging and opportunistic fungal infections. So emerging means every day by day, new fungal species are becoming, and they are becoming the opportunistic because first thing is climate change. So మనకి climate change లో being most affected కానీ లేకపోతే ఏదైనా global అదే every time temperatures increase అయిపోతున్నాయి because of the climate change also geographical change of the pathogenic fungi is increasing day by day and COVID-19 associated with the mycosis. So you all must knowing that there are so many COVID patients who died because of the black eye. So that is because of the mucormycosis black fungus and hospital acquired fungal infections because of the aspergillus or cancer infections are rising especially in the ICU and ventilator patients.
సార్ ఇక్కడ
The fourth reason why to study the fungal infections because lack of awareness, underdiagnosis, and delayed treatment. So, for the bacterial infections, diagnosis options are there, but for the fungal infections, there is no diagnosis factors and lack of awareness because everyone must be knowing if some fever comes or for some cold comes, immediately they'll give the blood sample for the test. But fungal infections, the blood low can come. So what means awareness రాకపోవడం వల్ల immediately we used to take care of the bacteria and viral infections. And delayed treatment because there are no fungal agents, antifungal agents, and no diagnostic test available for the fungal infection. Treatment must be is being delayed. And the fifth crops like the rice and wheat, which are the main components in our food, staple food. So in the rice blast disease and wheat blast disease, they lead to the significant economic loss and food security. So because of these reasons, study of food, fungal infections is important. So first we should know about the classification of the fungi. So fungi are so many types, you know, and by northwest appearance, fungi are divided into two classes: east like and molds. Yeast, they are similar to the prokaryote type. They are round in shape. Yeast like means they are both they exist in both the ways, like in round form and in hyper form. And molds are the long branching proteins. So the difference between yeast type and molds is that yeast type will contain hyphae without any septations, but the molds will contain two hyphae with septations. And another pattern is based on the aerial mycelium and vegetable mycelium. Aerial mycelium, because they grow on the surface of the culture or surface of the medium, so they are the aerial mycelium type, and vegetable mycelium type is submerged. They can grow inside the culture medium also. Type of fungi, they exist in both forms, like in round form and hyper form. Among the type of fungi, the examples are Histoplasma capsulatum, Blastomyces dermatitis, Coccidites and Paracoccidites and Penicillium coleoptile. So these are the types. Nothing is like yeast like and yeast except that hyper yeast like and other myces like dermatitis. And taxonomic classification. One must be studying like in zoology or botany. So what are the different types of different classes of the fungi? So one is the zygomycota, ascomycota, basidiomycota, and pteromycota. This is based on the types of spores with the flips. Laboratory diagnosis of fungal diseases. So how a fungal disease or a fungal infection can be diagnosed. So there are two main methods: microscopic method and macroscopic method. In the microscopic method, it depends like observation of the fungal species under the microscope. The sample collection depends on the site of the infection, like skin or scales or hair or sputum. Systemic mycosis. So they infect the inside of the body. So blood samples or cerebral spinal fluid, pulmonary fluid, or other types can be taken as a sample. So microscope, fungal infection under clinical microscope, based on the formation of the hyphen or body type. So the primary one is the treatment is potassium hydroxide. When the eight samples of skills which are the nails, they treated with 10% of chemotherapy. The human sample will be digested, only the fungal hyphae will remain and they can be checked under microscope. So there are other methods like gram staining, Indian ink, nitrogen stain, charcoal white stain, pathological stain, pyrotic acid stain, and others. Mason in California stain, Xenon, eosine, music carbon stain. All these stains can be used to identify the fungi in the culture. The main correct identification of the fungus is based on the macroscopic appearance, because we need not go for the microscopic identification of it. Because fungi, they will produce aerial mycelium, which are easily identified in the vision. So each fungus is having a different color. So for example, Aspergillus niger, they produce black color spores. Aspergillus niger have the parasite crop, it produces green color spores, and mucor, it produces almost bluish green color spores like that. So in terms of growth also, they are divided into two types: rapid growth kind of growth slow. Rapid growth is shown rising before the within areas. They are the main, they are the opposite mitosis, and slow growth is observed in 1 to 4 weeks. They are almost like they are prototypes for subcutaneous or systemic mitosis. So again, the identification is based on the nature of the hyphae, whether they are accepted
infection is the main, whenever we talk about fungal infection, we always know about candidiasis. So for candida, we only test may be geographic test may be connected to identify and coordinate from and for the fights the pay for test and previous result can be done for previous enzyme producing. Next, antibody detection tests like ELISA, immunodiffusion, or activation tests can be done for identification of and antigen detection like radius segregation or some for the fluids from surface molecule and immunohistochemistry for the tissue species. So the molecular mechanisms for identification of fungi are for PCR. In PCR also several modifications are there, multiplex PCR, tested PCR, RT-PCR, etc. And recently, very recently, automation identification systems like Molotov, VTech, Illumina sequencing, Nanopore sequencing, these are the DNA, latest DNA sequencing methods. Illumina sequencing reads the short fragments of DNA, whereas the Nanopore sequencing long fragments of DNA can be sequenced. And these are becoming revolutionary in accurate identification of the instant nodes. So now we will see what are the different types of fungal infections. Superficial mycosis means they are on the surface, like skin or hair. So the first one is Pinia versicolor. So this is caused by the lipophilic fungi characterized by the fat or scaly hypopigmented patches. The second one is Pinia nigra, characterized by painless black non-scaly patches. So two of the Pinia versicolor, XR1 and the right side figures are Pinia black color patches. So అన్ని అనుకుంటారు అంటే మీరు చదవకే కానీ ఇలా ప్రాసెస్ కానీ ఇలాంటి వాళ్ళకి అందిస్తే ఇమీడియట్లీ మనం ఎవరిని అప్రోచ్ అవుతాము బ్యూటీ పార్లర్స్ అప్రోచ్ అవుతాం ఎందుకు అంటే అవి డాన్స్ అనుకుంటారు సంథింగ్ ఏదో బట్ దే ఆర్ ద ఇన్ఫెక్షన్ ఆల్రెడీ సో మెడికల్ ఇది తెలుసుకుంటే ఏంటంటే పొద్దున మీ చుట్టుపక్కల కానీ సరౌండింగ్స్ ఎవరైనా ఇలాంటి కేసెస్ తీసుకుంటే వచ్చినప్పుడు advise them so to go for the doctor, to contact the doctor, so that then for children మొత్తం అంతా చేయొచ్చు. And next one, fever. So commonly అందరికీ ఉంది ఏంటి fever అంటే dandruff. Okay, so characterized by nodular formation of the facial. Okay, either black in color or multi color. So చాలా మంది చెప్తారు సో once ఒకసారి వచ్చింది after 6 months so better. Next, superficial mycosis in the dermal mycosis. The most common superficial mycosis affecting the skin, hair, and nails, caused by a group of related fungi called dermatophytes. So, dermatophytes, so examples can be found in Phytophyton, Mycosporus, and Epidermophyton. So, see, in fungal infections are simple to countryside, but when it grows, they lead to a drastic loss. So nails మీద ఉన్నాయి చూడండి హ్యాండ్స్ మీద పైన అట్లా ప్యాచెస్. నెక్స్ట్ సబ్క్యూటేనియస్. ఇప్పుడు దాకా మనం సూపర్ఫిషియల్ చూసాము, సబ్క్యూటేనియస్ అంటే అండర్లైన్ ద స్కిన్. సో మైస్కోమా. సో కామన్గా ఏంటంటే మగ్గ ఫుట్ అంటారు. సో మగ్గ ఫుట్ ఉన్న వాళ్ళకి ఏంటి ఫుట్ మీద అంతా సో ఆల్సో మన మదర్ మైకోసిస్ అండ్ మదర్ chronic slowly progressive granulomatous infection of the skin and stomach. So clinically manifested that so these are symptoms like swelling, discharging signs, and presence of damage in the viscera. So two types: blue mycoma and active mycoma. Blue mycoma is the true fungi, and active mycoma is false. So this is the mother of next other other one is so it is also known as both factors caused by pulmonary diagnosis, heat resistance. So for its skin, so it is present as a In the second one, so these are the various signs of health. Next, cystic mitosis. So it means inside of the body, inside the fluids, like fluids or cells. So it is caused by the transformative fungi. It's spread by inhalation. So inhalation will be caused by pulmonary infection. So transformative fungi examples include histoplasma capsulatum, plasmomyces dermatitis, toxicity, anaphylaxis. Next, first, so ఎక్కడ ఛాన్స్ వరకు అది స్టార్ట్ caused by fungi that are normally found as human organisms which depend on the human system or in the environment. They cannot pathogen low immune function. So that's the immunity maintenance is very important. So the first one comes the candidiasis. So it is a common fungal disease. So it affects skin, mucosa, and various microorganisms. Caused by Candida, species include Candida albicans, Candida botanis, Candida Gabrata. There are so many different species. Common in the presence of various predisposing factors, patients with steroids or immunosuppressive drugs. This is one because of the Candida infection. So this is the oral candidiasis. So either check and find on the first oral candida test, and second one is male candidiasis. Into the visual circular candidiasis, candida vaginis and so Cryptococcus. Cryptococcus is caused by encapsulated yeast-like Cryptococcus pseudomonas capable of producing fatal meningitis in sexually people. So next is zygomycosis. It is a life-threatening infection caused by the insect fungi Zygomycota. They include the viruses, mucors, other risk factors in diabetic retinopathy. Simple forms include rhinocerebral mucormycosis. So see, and e patient in the left side patient that is the abdominal region. He went to an oil massage for a month. So the fungi infected from the oil. So it infected from okay. So that's why the internal organ, whole internal around the liver, everything recording. So that is the cytomicron. And then aspergillosis. So first we enter into allergic disease caused by the hydrogen and aspergillus. Aspergillus చాలా types ఉన్నాయి species. Aspergillus niger, Aspergillus tincti, Aspergillus parasiticus, Gravus, Aspergillus niveum, etc. So important species indicators levels. Risk factors are glucocorticoids and contain so simple forms include pulmonary aspergillosis more common, and other forms like sinusitis, cardiac, severe or ocular arthritis. So ocular and eye infections are explained. Actually, ఏంటంటే ఫంగస్ గురించి so this is a aspergillus species. So inside the mouth also there and all the skin So microsporidia are transitory declassified fungi. So it is not a fungi, but the fungus is fungi. So they comprise several species. Example, Elsevier rubulon and which causes the gastrointestinal musculoskeletal and ocular infections. Main question is microsporidia is for ocular infections. So ICO మరియు కొన్ని సార్లు బ్లడ్ కండిషన్స్ అయితే వాటితో మనం and then treatment options. What are the treatment options available for this fungal infection? The main thing, as I told you earlier, polymers, chemotherapy, and endophthalmology. Ivanni ఈ three types మాత్రమే ఉన్నాయి. So వాటిలో drugs, medicine, etc. So what we uses and what we can the value of this the main ఏంటంటే బ్రేక్ చేసే డిస్టర్బ్ చేసే ఆల్కహాల్ ఏజెంట్ అని చెప్పి నెక్స్ట్ ఫంగల్ కంటెంట్స్ బీటా గ్లూకాన్ సో బీటా గ్లూకాన్ inhibiting agents are there and mitotics will bind to the fungal cell wall. So these are the agents but they are not that much and the other one is azoles. So azoles help in this fungal infection but up to the so what is the current scenario of fungal infection in the market? So what is the scope for working on fungal infections? So one is the global prevalence. So what's the global prevalence of fungal infection? Nearly 1 billion individuals suffer from this aid and tail fungal infections. Tens of millions they experience something new or mutable malnutrition. Over 150 million people suffer from serious fungal infections. And the invasive fungal infections, they range from 6.5 million cases annually with 3.8 million deaths because of that. And then the impact of the climate change. The first is the second slide we have noted that climate change also leads to fungal infections. Climate change is already fungal infections. And because of the is a one which is expanding to the new regions of the Canada soil. So in Canada, this Pickle is definitely not protected. But because of the climate change, it is expanding into the fungi are adapting to their temperatures. One thing we have to make a note that bacterial infections or viral infections they can survive only at 30 but fungi they are omnivorous and omnivorous. Means they can grow at 4 degrees, they can grow at 20 degrees, they can grow at 25. There is a so because of it. Next, increasing concerns of fungal infection because increasing the antifungal therapies. Only few options are available as an antifungal. So they have become resistant to the vaccine. And then emerging opportunities fungal infections like candida is growing day by day. And in immunocompromised patients like COVID as so that the fungus that fungus so that is only immunocompromised in the COVID-19 patients. And global health organizations using better harvesting. The global health initiatives. So what are the initiatives that are taken so far for infections like 95 by 25 means they aim to diagnose 95% of the fungal infections and treat 95% of them by 2025. And copy Global Action Fund for fungal infections. It is linked to global efforts. And then increase funding and collaborations. So usually in bacterial infections production disease and viral disease and we get the funding grant in form of grants from ESP or EU etc. But fungal infections they are not given that much importance. So nowadays the government is also concentrating on giving funds to fungal infection studies also. And antifungal drugs, the market overview. So by 2022, the market value for the antifungal infections with antifungal agents is 15.8 billion US dollars, and the projected growth is up to 19.6 by 2030. And another study also reported the same thing. By 2030, the antifungal market growth will rise to 20.6 billion US dollars, and it is because of the increasing fungal antifungal, what are the different types of fungal infections and how to treat them using the antifungal agents. But there are several alternative methods to be improved. So alternative strategies to prevent and treat the fungal infection beyond the antifungal agents are: one is, first one comes the enhancing host immunity. So always we should develop immune system. So by in COVID-19 patients, immune compromisation is there. So they have to undergo immunotherapy. So boosting the body's immune response will help to fight against the fungal infection. Second one is cytotherapy, like interleukin 17 or interference gamma. So their uses can enhance the antifungal immunity. Then dendritic cell therapy. So modulating the immune response to enhance the fungal resistance will also is an alternative. And the vaccines. So we have seen the viral vaccines or bacterial vaccines, but now the fungal vaccines for fungal infections is also under development. For example, Candida vaccine, NDV3A, targeting the Candida. So once the vaccine comes into the action, then it can be taken at the lower age group. I mean, one polio virus is అదే నెక్స్ట్ cervical vaccine, cervical cancer vaccines we are taking at the age of 14 to 16 years. So like that only, for Canada also, if you take the vaccine before the adolescence, there is a less chance to get the next one, cryptococcus vaccine. So live attenuated cryptococcus neoformal strain is used as a vaccine. Next, aspergillus vaccine is also under development, preventing invasive aspergillus, and antifungal antigens for the neurospora. Recently, we have submitted a proposal since the neurospora classes are harmless fungus. So, neurospora is a fungus which is eaten, which is edible in Russian countries. So, this is a harmful fungi. So, I am working on this fungus since 2 decades, since 1999, since my PhD, I am working on this. So, from this neurospora classa, the antiviral sequence are there, antibacterial virus are there, sequence are there, and antifungal sequence are there. So those sequences we wanted to identify, and we want to develop antidotes for the infections. So this proposal we have submitted recently to ICMR. And use of probiotics. So beneficial microorganisms. We know that secondary lactobacillus is important. So instead of these probiotic bacteria, we will also inhibit the growth of the fungal pathogens, particularly Candida. And probiotics can restore the gut and vaginal microbiota. And other treatment option is fecal microbiota transplantation. So మన గట్లో ఏవైతే యూస్ఫుల్ మైక్రోబ్స్ ఉంటాయో వాటిని ఫీకస్ ఐసోలేట్ చేసి వాటిని క్లీన్ చేసిన తర్వాత మళ్ళీ ఒక ఆప్షన్ ట్రీట్మెంట్ ఆప్షన్ కింద యూస్ చేయండి. సో దిస్ ఈస్ అండ్ ఆన్గోయింగ్ ఇన్ యూఎస్ and then phytochemicals and natural antifungal components. So the treatment options for these antifungal agents all are chemically similar. So by taking the phytochemicals or natural agents from the plants or other components, they also serve as an option. So plant-derived essential oils and phytochemicals, like eucalyptus oil, rosemary oil, and lemongrass oil, So all those oils can be taken as the option. So PPA oil, thymol, eugenol, and curcumin, they have shown some antifungal properties to Candida and aspergillus. So and Siktor and Geranium from the recent study, we only published in Regulon in 2022. So these two oils will also serve as the antifungal agents. Then marine natural products. So marine natural products means marine animal samples from sponges and seaweeds. So natural products from these sponges or seaweeds, they can be derived and they can be used as antifungal agents. Nanobiotechnology based approaches. So nanobiotechnology is on a last decade research nanotechnology. So production of the nanoparticles, nanoparticles or something else. So use of this nanotechnology also an option for the antifungal side. So single nanoparticles or hydrogen nanoparticles and ribosomal they are found to be bacterial and antifungal. And zinc oxide and nitrogen oxide nanoparticles, there is it is also reported. And liposomal is formed to improve the drug penetration and reduction in the toxicity of fungus. CRISPR, CRISPR-Cas9, so where it is used in treating the obesity, first discovered. So the same technique, the same medicine that we use as antifungal resistance. CRISPR-Cas9 gene editing tool to target fungus. So using the fungal virulent genes, the fungal pathogens. So those genes has to be identified and with the gene editing tools they can be removed. So this is also we have submitted a project and genetically modifying beneficial bacteria with antifungal fungus. The preventive strategies and environmental control. So what are the preventive strategies? What we can do? Like we have to improve the hygiene and personal care and then decontamination and biosecurity. So hospitals are encouragement you have to have wash your hands, you have to take proper care, infiltration, and hospital and then fungal infection and surveillance and healthy diets. Whenever you come across a small itching should not immediately go to diagnosis and like other things for diagnosis. So with the help of the improve biosensors and rapid molecular that is fungal infection early stage. So coming to the conclusion, before I go to the conclusion, you want to show another slide. So we are working on this. So she's a lady, she's a 7 years age. The ఏంటంటే this situation ఏంటంటే ఈ ప్రాపర్టీ నెయిల్స్ కానీ హెయిర్ చూస్తే లేకపోతే ఆల్వేస్ ఏంటంటే డిశ్చార్జ్ సో ప్రాపర్టీ బాగా సివియర్ ఇష్యూ హాస్పిటల్ తీసుకొని వచ్చినప్పుడు డయాగ్నోస్ చేస్తే వీళ్ళ పాప మదర్ రాసిన దానిలో ఇన్ఫెక్షన్ బికాస్ ఆఫ్ విచ్ షీ బౌండ్ లైక్ దిస్. So we have done a preliminary study. So అందుకే కంప్లీట్ గా చూపించలేదు. సో దీని ఫస్ట్ దీని మీద ఐడెంటిఫికేషన్ చేయడం ఇవన్నీ జరిగింది. అండ్ కమింగ్ టు ద ఎగ్జాక్ట్ ఐడెంటిఫికేషన్ ఆఫ్ ఫంగస్ అండ్ మాలిక్యులర్ టూల్స్ స్పీషీస్ ఏదైనా చేస్తున్నారు. ట్రై టు ఫోకస్. సో క్లాసింగ్ దాంట్లో ఉంది అంటే మన మదర్ని చేయడం కదా. అంటే ఫస్ట్ డైలీ హెయిర్ వర్క్ చేస్తాము, డైలీ ఇష్యూస్ అని అన్ని తీసుకొని వర్క్ చేస్తాము. సో this is how dangerous the fungal infection అనేది చూపించడం కోసం వచ్చారు. So coming to the conclusion, fungal infections range from mild infections to life-threatening stages. So proper classification and this is important. Climate change and natural hygiene are the main reasons for the increase of the fungal drug resistance also is a cause. So antifungal drug market is expanding and new drug development is necessary. Early diagnosis, awareness and improved treatments are important to control the fungal infections. So the take-home message for this is beyond the antifungal drugs, immunotherapy, probiotics, natural compounds, nanotechnology, genetic engineering, and preventive measures. They are crucial for controlling the fungal infection. So multidisciplinary approach like combining any of these 2 or 3 techniques will definitely improve the treatment efficacy and to reduce and enhance the public health awareness.
Thank you so much, ma'am, for the wonderful lecture that you have created a lot of awareness among us how fungi can affect us and how we also need to keep ourselves in a healthy, pollution-free environment. Dear students, the floor is open for queries. If any one of you would like to ask any questions or clarification, you can do so. You are most welcome.
Excellent presentation, ma'am. Thank you very much for coming over subject, aged 32 years, and he is suffering from congenital hemophilia. During 2020, that is COVID-19 time, and he was affected with the COVID-19, and he was admitted in that hospital, Hyderabad hospital, and that's a government hospital. It's completely on oxygen ventilation, and he was attacked with a mucormycosis. Already mentioned, is this black fungus, and he was infected with that. Very unfortunately and badly, he lost his eyeball, and he lost his upper jaw. And again, due to this hemophilia, and we were unable to do the operation to reconstruct that upper jaw and maxillofacial surgery reconstruction, and it was not possible. Still, he's leading his life as a great warrior. He's lost the life of unable to eat also. See, when you are presenting all those slides and recapitulating his life history, how he has passed all those things, how he is leading that life. Excellent, ma'am, excellent. Thank you very much for coming over here. Thank you. Good morning, ma'am. Anything we can give it from plant extract like retail neem extract because tea tree oil and other oils are very expensive. So anything very easily available can make antiviral at home like garlic extract, neem paste, something like that is available. You can do the what neem yeah that we can just because only call they are available in small yeah it is again them like the copy very cost yeah but they are advertisement where they are available is how we can prevent not to get fungal infection or body needs things. Something that can with soil and keep your nice feet for some 10 or 15 minutes like that. Easy work for that. Sometimes what to say marine soil they are they are quite useful. But the thing is that we have to cut out things regularly. So sometimes the passion or something like that. We can use them for the days. And even a small microscopic particle at least weekly once I have yeah, personal health is very important. But we have to make aeration. Aeration have to maintain aeration. So always we have to make sure of our hands and everything are right. Thank you. Thank you so much.
Anyone is there to ask questions? Thank you. Ma'am, I'm grateful to you for being with us and enlightening each one of us about the fungi, its structure, morphology, the types, and the diseases caused by fungi. Listening to it was frightening. And he also said once we get infected, we cannot, there is no cure. Like so, and also told us that how even climate also is playing havoc in fungi diseases. Also, thanks for sharing with us the various treatments that are available for fungi diseases, giving us hope even to educate people those who are affected by that they should not be disheartened, but there is still cure for the particular disease. And also surprised to see once you listen to you, they are able to adapt themselves according to the climatic condition, even extreme this one temperatures. Okay.
Like a host or plant or animal. You know that. Outside of the medium, it has a particle with part. It is non-living. But for bacteria means it like only at 37 degrees. Whereas the fungal spores, zygospores or polyospores or any other bacterial spores, any kind of spores, they live for at least 25 years, more than 50 years, without any culture medium or anything. They are scattered in the environment, and whenever they find any favorable space, they will try to develop. Like, you'll be knowing like whenever the sugarcane cultivation is good, they will what they will do? They will burn it. So after burning also, the neurospora sections then only develop. So again, it is a chance. Even at 120 degrees, the burn medium, the burn temperature cannot affect this. So that is the survival capacity of the fungus, even at the sporulation stage, not at the mycelium stage. Okay, it is completely adverse, negative, reverse to humans. Like infants, they cannot survive without any support or food. Only adults can survive, that too to some extent. But whereas the fungal spores, the infant stage, they can survive more than 25 years without any medium.
Thank you, ma'am. Also telling us to develop good immunity. The students, it is for you especially, your youngsters, you need to build up your immune system well and also have a healthy hygiene to develop for you so that even avoid the pollution um, areas where you also can get, there is a possibility for you to get it. Ma'am, thank you for telling us that and also telling us also to be careful and create pollution free environment. Thank you so much, ma'am. I would like to extend a heartfelt gratitude to our esteemed invited speaker, Dr. J. Navina Lavanyalata, for her revelatory and reflective address. Your proficiency, profound observations have augmented our understanding on fungal infections, classification, diagnosis, and management. Thank you, madam, for taking the time to share your knowledge with us. I would also like to express our sincere gratitude to our co-chairperson, Dr. Sister Japmani, for your invaluable support and guidance throughout this session. Your contributions have been paramount in making this session a success. Thank you, Sister, for your dedication and commitment. It is now my honor to invite Dr. Sister Japmani to felicitate Dr. J. Navina Lavanyalata with a memento as a gesture of gratitude for her invaluable contribution and insightful talk. Dr. Sister Japmali, may I kindly request you to come forward and present the memento to Dr. Navina Lavanya Lakana. Thank you, ma'am, and thank you, sister. I kindly request you to occupy the chairs of the dance. We would like to invite the dignitaries and participants to take a short tea break. We will resume in 15 minutes for the oral session 2. Dear participants, the refreshments are available outside the conference hall. We request everyone to return and be seated by 11:30 a.m. The oral session 2 will be held here in the conference hall itself. Thank you.
హలో బాబు ఇది పార్టిసిపేట్ వైట్ సైడ్ పెట్టింది స్టేజ్ అది హ్యాండ్ బ్యాగ్ పెట్టినప్పుడు రావట్లేదండి అక్కడ వినోద్ వినోద్ లేడు స్టేజ్ కలర్ నేను ఇక్కడ ఉన్నాను గ్రీన్ కలర్ అంటే వన్ 102లో ఏది గ్రీన్ కలర్ కిందదా ఆర్చ్ 1 2 3 4 ఉన్నాయి 1 2 సారా
Sciences, Oxford University, Oxford,
1, 2, 3, 4, 5.
Ethiopia to deliver the invited talk virtually. Sir, may I kindly request you to proceed with your talk?
Huh?
Hello, sir. No, not sir.
Where is it? Where's the picture?
ఆయన అన్మ్యూట్ చేస్తున్నారు సార్. మ్యూట్ చేయాలి.
It is audible? Yeah, it is audible?
Yes sir, yes sir.
Yeah, sir. PPT is sharing, sir?
Yeah, 1 minute sir, 1 minute.
Okay. Sir, it is shared, sir?
Sir, is it okay?
Yeah.
Yeah, okay sir, you can proceed sir.
Okay, sir. Okay. Sir, it is moving? It is
Yeah,
moving?
yes sir.
Moving? Okay. Yeah, thank you very much for the invitees who invited me to present my presentation on which is very relevant to your conference, the integration of artificial intelligence and green technology for sustainable treatment of inflammatory as well as breast cancer. Okay. Before presentation, myself, I am Dr. Krishna Chaitanya, working as Associate Professor in Oxford University since 15 years. I'm having wide experience in the field of the biochemistry, biotechnology, microbiology, at teaching as well as research and community service project. Now, I will start my presentation. Okay. You see my presentation having the following outlines. First of all, introduction, statement of the problem, objectives, materials and methods, results and discussion, as well as conclusion. You see, before going to that, you see, my title is, yeah, the full, I kept the full screen.
Is it okay sir?
Yeah.
Okay.
Sir, okay? It is moving, sir? It is moving? Yeah. Sir, slides are moving?
Sir, I can operate this one.
Okay, you can operate. Okay,
Yes.
right. Okay. Yes, first of all, I'm going for introduction part. Before going to the introduction part, green technology or the biotechnology is the field which deals with integration of biology with the nanotechnology, okay, which deals with development of novel drugs by using nanotechnology. Nanotechnology is a broad field which deals with different properties like the physical, chemical properties, like that. You see, that's why green technology, the novel science which deals with the development of the novel nanoparticle with a different physico-chemical property. Okay. In green technology, we are using natural resources like the plant product as well as the microorganisms for the production of nanotechnology. these nanoparticles which are having anti-inflammatory, anti-cancer, antimicrobial activity. Okay, simply green nanotechnology is a science which is emerged as a new broad science of the nanotechnology for the development and the treatment of the novel medicines. Okay, recently scientists they focus on different metals and metal oxides. Already the metals and metal oxides like silver, gold, zinc oxide. Already they proved that they're having the provenant pharmacological activities such as antibacterial, antimicrobial, or anti-inflammatory, anti-cancer activity. That's why these metals are having the profound activity. We can enhance the profound activity of the metal by combination of the bioactive compound which is isolated from the plant extract. That's why now our major aim is the development of the nano medicine. Nano, nano means the size of the nanoparticles are 1 into 10 to the power of minus 12. That's why now our major focus on the development of the nano medicine by using the green nanotechnology for the development of the drugs, for the drug delivery, imaging, diagnosis, and identification of the diseases and everything. Okay. You see, nanoparticle. Before going to that, what is the meaning of the nanoparticle? Nanoparticles are the molecule which may be organic or inorganic or biological molecule which are having the size of 10 to the power of minus 9 nanometer. But these nanoparticles having the different properties and a different biological important. These nanoparticles are prepared by different methods like the physical, chemical, as well as the biological methods. But among the various and physical methods, they're having their own disadvantages. That's why now we are focusing on the preparation of the nanoparticle by using the green method. In the green synthesis, we are using the plant extracts as well as the microorganism as well as the fungi. Why we select the green synthesis? The green synthesis is eco-friendly and we no need to use cost-effective instrument. That's why when compared to various physical and chemical method and biological methods which can use low equipment method and we cannot use any hazardous chemicals. That's why nowadays scientists they focus on the synthesis of various metal and metal oxide nanoparticle by using the principles of green chemistry, which is eco-friendly nature and as well as we can control the synthesis of the nanoparticle with the less time and which improved physico-chemical properties with potential biological activity. Okay. As I told, nanoparticles, what do you mean of nanoparticles? Means a group of the molecule which may be organic or inorganic or the biological, which are having the size of 10 to the power of 9 nanometer, which can be synthesized with the three different methods. One is physical, chemical, as well as biological. As I told you before, the physical method having its own more disadvantage. That's why the scientists they focus on the novel method which cannot have any harmness to the human being. That's why the scientists developed a new method. It is called green synthesis method. During green synthesis method, what is the meaning of green? You know very well. Green means due to the pigments of the chlorophyll A, chlorophyll B, chlorophyll C. The term green is coming. And in green synthesis, we are using plant, bacteria, fungi, yeast, and viruses because it is a simple, non-toxic, economically, and eco-friendly. Why we are selecting green? Because you saw when you see green, when you see traditionally all plants, traditionally medicinal plants are useful for the treatment of the different diseases due to presence of the secondary metabolite. That's why every plant having its own significance for the treatment of the diseases due to presence of the secondary metabolite. You know very well the penicillin is the antibiotic which can be discovered by the fungus. That's why the fungus, as may be the plant, it contains the different secondary metabolite. Those different secondary metabolite which are having the biological activity. Okay, that's why I'm focused on the synthesis of the green method. Okay, in green, we are using the plant, fungi, yeast, and viruses. But I selected the plant. Why I selected the plant means the cultivation of the bacteria, fungi, yeast is it is very difficult and cost effective also. That's why I'm focusing on the synthesis of the nanoparticle by using the plant. Why? The plant containing the secondary metabolite. These secondary metabolite which are having biological activities, different biological activities, as well as these secondary metabolites also responsible for the synthesis of a nanoparticle, which act as reducing agent, capping agent, as well as what? Stabilizing agent. That's why What is the meaning of green synthesis? Green synthesis is the method in which the preparation of the nanoparticle by using the biological agent. Among the biological agent, the plants are very simple, eco-friendly, eco-friendly and easy available living organism which contain the secondary metabolite. These secondary metabolite having the dual activity. One is it acts as reducing agent, capping agent, as well as stabilizing agent, as well as it is also having the biological activity. Okay, that's why you see the different scientists already reported that the phytocompound which present in the plant extract having all these activities. That's why my important point here is development of novel anti-inflammatory nanomedicine for the treatment of cancer. For the treatment of the cancer, I didn't include it here due to the lack of due to the limitation of the slides. Nowadays we are having so many different cancers are there. Among the cancers, the breast cancer in the females occupy the large mortality rate or huge mortality rate in females. That's why so many breast cancers are there. Among the breast cancer, recently scientists 5 years before they identified the inflammatory breast cancer. Inflammatory breast cancer. You see, the inflammatory breast cancer is the breast cancer which is due to activation of inflammatory mediators. That's why my objective is development of novel anti-inflammatory agent from the medicinal plants, as well as for development of the nanoparticle by using the plant extract for the treatment of inflammatory breast cancer. That's why. What is the meaning of breast cancer? You know very well. Breast cancer is the type of the infection. It is uncontrolled growth of the breast. Among the so many types are there, but inflammatory breast cancer is a more aggressive breast cancer. Nowadays, it is activated. It is activated due to the different causes like our food habit, as well as the smoking habits, as well as like daily food habits. That's why you see now my consideration is development of novel anti-inflammatory agent from the metal and metal oxide by using the plant extract for the controlling of what? Inflammatory related breast cancer. That's why nowadays scientists they're using the different anti-inflammatory agent for the treatment of the breast cancer, but they're having their own limitations. So they're having their own limitation. That's why I want to develop the anti-inflammatory molecule which does not have more cytotoxicity on a human being. That's why already you know very well different steroidal as well as non-steroidal anti-inflammatory agents are there for the treatment of the breast inflammatory related breast cancer. Okay, here very important. You see, green nanotechnology is the combination of the synthesis of nanoparticle by using green sources like medicinal plants. But here the synthesis of the nanoparticle, already as I told you, nanoparticles are synthesized by 3 different methods: physical, chemical, as well as biological. Among the biological, plant mediated green synthesis of the nanoparticles are very easy and cost effective. But here important thing is now when AI is developed, we no need to do more work because already so many scientists they published the different data on green synthesis of the nanoparticle by using AI tool. AI in machine learning, 2 different type of the tools are there. One is supervised, another one is called what? Non-supervised tool. Means supervised AI tool, another one is called what? Unsupervised learning tool. That's why you see nowadays, for example, recently since 1 year, I am focusing on synthesis of the nanoparticle by using artificial intelligence. In artificial intelligence, machine learning is there. The machine learning, the 2 different type of machine learnings are there. One is supervised machine learning, another one is called what? Unsupervised machine learning. In supervised machine learning, with help of the deep learning, with help of the artificial intelligence, already exist based from the existed data, it can give some idea how to prepare the nanoparticle, how to optimize the synthesis of the nanoparticle and everything. Another important thing is also there, unsupervised learning. Here very important. One is already known. Supervised means we know the data, based upon the data, we synthesize the nanoparticle. But another one is unsupervised learning also there. These unsupervised learning, we don't have any data, but AI, it can create its own data based upon our own output. That's why nowadays the scientists they synthesized the novel nanoparticle by using the green chemistry. Nowadays it is very easy by integration with what? AI. Another thing, you see, say example, when you see the slide, one is supervised learning strategies of development of novel nanoparticle. Another one is called what? Unsupervised learning model. Okay, you see machine learning. In machine learning, in machine learning, two different things are there. One is supervised learning. Another one is called what? Unsupervised learning. Okay, you see, in green synthesis of the nanoparticle, having the 5 different major steps are there. In the major step, first we have to select our plant extract. Okay, after selection of the plant extract, we can go for the extraction. After go for the extraction, we identified some secondary metabolite. These secondary metabolites are responsible for the preparation of the nanoparticle, which act as stabilizing, capping, as well as reducing agent. That's why you see already the scientists they identified they developed different nanoparticle by using the medicinal plant. By using the supervised machine learning process, already it can create the data. These secondary metabolite like the flavonoid, terpenoid, already the different scientists identified, which is having the more responsible for the reducing agent, capping agent, as well as stabilizing agent. That's why by using the machine learning model, we have to optimize the synthesis of the nanoparticle by stabilizing the pH, temperature. You see, generally conventional methods. For example, while the preparation of the nanoparticle, first of all, we select the plant. We have to select the plant, then extraction, then identification of the phytocompound. Then after identification of the phytocompound, we can mix metal or metal oxide with our plant extract. Then we can do for optimization. Optimization means we have to affect the pH, temperature, everything we have to check. But nowadays, when AI is integrated, we don't need to do everything. For example, for doing of this work, it will take nearly 3 months like that. That's why when AI tools are developed within one, not more than 1.5, not more than 30 minutes, it can optimize itself. Which pH is optimum pH is responsible for the synthesis of the nanoparticle, which temperature is necessary for the synthesis of the nanoparticle. Everything it can give its own predictions. Second one is based upon the supervised learning, we can get our control size of the nanoparticle. Nanoparticle having the different size according to our interest. For example, breast cancer treatment, the important thing is we need 20 to 30 nanometers of the size, and shape spherical is we need spherical size, and nanometer size also 20 to 30. That's why by using AA, it is very simple. It is very simple to predict the output. Okay, that's why we no need to go for optimization. By using the AI, it can do itself, and we can get our desired size of nanoparticle with more stability and more yield. Another thing also, we can identify the bioactive compound which is necessary for the stabilizing and capping agent. Okay, you see nanoparticle. Okay, for example, after the synthesis of the nanoparticle, the conformation of the nanoparticle is very important. You see, conformation of nanoparticle is very important. That's why in nanobiotechnology, by using the different instruments like a spectroscopic instrument and a microscopic instrument, we can confirm the nanoparticle size and the nanoparticle physicochemical properties by using like UV, FTIR, XRD, SEM, TEM, EDX, DLS. Among these spectrophoto properties, UV, FTIR, XRD, these spectrophoto properties responsible for the confirmation of which metal is responsible for the synthesis of the nanoparticle, and microscopic techniques which is also useful for it can measure the size of the nanoparticle and the morphology of the nanoparticle, everything. And EDX means which metals are present in our nanoparticle, and the DLS, very very important instrument, the DLS instrument which can give the random size of the nanoparticle as well as what surface area. That's why I already mentioned here the characteristics of the nanoparticle done by two different instruments. One is spectroscopic as well as what? Microscopic instrument. These two instruments which can confirm the physico-chemical properties of nanoparticle. But here you see this is not modernized method. This is generalized method. We have to do everything. But when AA is developed, we don't need to do everything, because already the scientists they published a huge amount of the data on the green synthesis of the nanoparticle. That's why when we integrated with the AA, it can collect all the data. Data is available. All the data is available about green synthesis of the nanoparticle and the characteristics of the nanoparticle. That's why what it will do when integrated with when we can give the command in the AA by using the machine learning, it can collect all related information. Okay. For example, what the conformation of silver nanoparticle, for example 400 nanometer, it can generate its own it can generate information based upon their pre-existing data. That's why nowadays for example before sending the sample for the characterization, we can predict nanoparticle size, shape, morphology, physicochemical property by using what? AA. That's why nowadays we are having the special mission tool, AA based spectroscopic analysis and AA based microscopic analysis and AA based distribution of the nanoparticle. By using the automatic mission, before sending the sample, we can predict our physicochemical properties of nanoparticles. Okay, you see, this is very important. That's why here introduction I mentioned three important things. What is nanoparticle and how nanoparticles synthesized? What are the disadvantages of physical method, chemical method, and what are the advantage of biological method? In biological method and plant mediated nanoparticles are very simple and easy method. But while the synthesis of these nanoparticles, it can take more time for selection of the plant extract, for identification of the secondary metabolite, to check the concentration of the molarity of the metal oxide nanoparticle. This is a normal manual method. But when we integrated with AA, we don't need to do everything. We don't need to do all traditional methodology. It can give. Okay, this pH is responsible for the synthesis of nanoparticle. Everything it can do everything. That's why here in introduction I focused on how AA is responsible for optimization of the nanoparticle and the characterization of the nanoparticle without doing a manual method. Now, my interest is statement of the problem. In the statement, very well. Already I mentioned about the inflammatory related diseases. You see, very well immunology. In immunology, the broad science of the science which deals with protection against diseases. We are having the two different type of immunities are there. One is innate immunity, another one is called what? Adaptive immunity. But in innate immunity, inflammation is the one type of the barrier. Okay, this inflammatory barrier is responsible for the prevention and the spreading of the diseases. But inflammation, two different types are there. One is acute inflammation, the chronic inflammation. Acute inflammation is the protective, but chronic inflammation is not a protective. But when acute inflammation is prolonged, it became the chronic inflammation. This chronic inflammation which can causes inflammatory related diseases. Some of the inflammatory related diseases are the breast cancer is one of the important inflammatory related diseases. Is called inflammatory breast cancer. In inflammatory breast cancer, all inflammatory mediators are upregulated. It can causes different. It can causes activation of different inflammatory pathway. These inflammatory pathways which can activate the growth factor. These growth factor which can responsible for upregulation of the cell cycle, and it can mimic the cell controlling pathway. It can progression of the breast cancer. That's why here during inflammation, all pro-inflammatory cytokines are upregulated. When pro-inflammatory cytokines are upregulated, it can activate the cancer. When it activates the cancer, automatically it can progress the cancer. But our aim is we have to develop the, first of all, we have to develop the novel anti-cancer agent by using the metal oxides as well as the plant textiles. This is called green sense of the nanotechnology for controlling inflammatory related breast cancer. That's why here why we are focusing on development of novel anti-inflammatory agent. Already in market, so many anti-inflammatory agents are there, but they're having their own disadvantages. When we are using the plant extract, it does not have any cytotoxicity. That's why our major aim is we are preparing the nanoparticle by using the plant extracts. Okay, that's why you see already different different anti-inflammatory agent, they're having their own disadvantages. That's why we are focusing on the synthesis of the novel nanoparticle by using the plant extracts for controlling of the cancer. Okay, but here important thing is the scientists up to now they didn't focus on development of more anti-inflammatory green sensitized nanoparticle for the treatment of inflammatory related breast cancer. That is a novelty here. That's why critical gap is development of novel anti-inflammatory nano medicine by using the plant extracts. Here, combination of three different sciences. One is immunology, pharmacology, nanotechnology. Now, AI also added. Okay, and so now, the current study is focusing on development of the novel anti-inflammatory agent by using plant extract as well as what metals. Here, integration of nanotechnology with the plant, medicinal plants, is responsible for development of novel nanoparticle because it having reduced cytotoxicity, lower dose, enhanced bioavailability, higher specificity, and a greater conventional system. Therefore, the current study is responsible for the development of the nanoparticle. Here, in my study, I prepared the two different nanoparticle. One is gold nanoparticle, another one is titanium oxide nanoparticle by using the solanum plant extract. Okay, for development of anti-inflammatory activity. You see, that's why. But you see, this is previously in 2022. I done manually, but when AA is developed, I don't need to do more work because already I checked with AA. When my results are when my previous results are compared with AA, it can optimize everything. You see, AA is responsible for the cell viability and morphological studies. AA is responsible for cytokine as well as what gene expression studies. That's why while development of the novel nanoparticle by using AA, it is very simple. We don't need to we don't need to do much. You don't need to spend much time on to development to search literature review because it can give everything by using the AA. That's why I'm now currently I'm using AA for to checking anti-inflammatory activity as well as to checking gene expression studies as well as to checking anti-cancer activities in breast cancer. Okay, my major objective is I am synthesizing two nanoparticle, gold as well as titanium dioxide nanoparticle by using the plant extract to evaluate anti-inflammatory activity. Okay, this is the specific objective. In the specific objective, I synthesized two different nanoparticle because why I selected two different nanoparticle is it is based upon the review of literature. Up to now, no one synthesized gold nanoparticle and titanium dioxide nanoparticle to targeting inflammatory related breast cancer. That's why I selected these two nanoparticles. Another important thing is also there. For example, nanotechnology not only useful for the development of the drug, nanotechnology having three important criteria. One is diagnosis of the diseases, another thing treatment of the diseases, another one monitoring of the diseases. That's why for the diagnosis, treatment, monitoring of the diseases, we need nanochip. Now currently, the scientists they're focusing on these two metals. One is gold, another one is titanium dioxide for the synthesis of biosensors and synthesis of the nanomedicine by monitoring cancer as well as what inflammation. That's why my first objective is I'm synthesizing here two nanoparticle, gold as well as titanium dioxide nanoparticle in green approach. Second one is I want to identify the which secondary metabolites are responsible for the synthesis of the nanoparticle, and after that I can go for the spectroscopic method to confirm the nanoparticle. I can go for the microscopic method to confirm the size, shape, morphology of the nanoparticle. Then after the synthesis of the nanoparticle, I can check cytotoxicity. Because here, why I selected, for example, when you see the fourth objective in assessment of cytotoxicity, SM means plant extract, AQ means aqueous, SM means plant, AU gold nanoparticle, titanium dioxide nanoparticle. Here, I prepared two nanoparticle by using aqueous extract. Now, I want to evaluate these two nanoparticle, among these two nanoparticle, which nanoparticle, you know very well, nanoparticles having its own disadvantage of cytotoxicity. That's why nowadays by using AI, we can optimize the cytotoxicity of what? Nanoparticles also. That's why here I prepare the 2 nanoparticle by using the aqueous extract of SM, solanum malgitanium. And first of all, I want to check the nanoparticle cytotoxicity. And after the checking the cytotoxicity, I want to screen anti-inflammatory activity. After the screening anti-inflammatory activity, and based upon the output of the result, I can check inflammatory induced breast cancer anti-cancer activities by using real-time PCR. Okay, the significance of result also there. For example, this is my background of the study. When you see thing, the first one, materials and method. First step is collection of the plant material and authentication. And after the, I selected here Solanum malginatum. This Solanum malginatum, after identification, because how selected this plant based upon the traditional important. After the selecting the medicinal plant, I powdered the medicinal plant, then I make into the aqueous extraction. After aqueous extraction, I can go for the phytochemical analysis. After phytochemical analysis, I selected the plant extract for the green synthesis of the nanoparticle, gold as well as the titanium dioxide nanoparticle. After synthesis of the nanoparticle, I confirmed the nanoparticle by using the both spectroscopic and microscopic technique. After the confirmation of the nanoparticle, I checked the cytotoxic effect of these two nanoparticles by using Russell. After the checking the this cytotoxicity effect, I optimize the concentration of these two nanoparticle, and after the optimization of these two nanoparticle, I checked with anti-inflammatory activity by checking the inhibitory effect on nitric oxide production. Nitric oxide is one of the inflammatory marker which can, as well as anti-cancer marker also. When nitric oxide levels are decreased, automatically it is showing anti-cancer activity, and after preliminary activity, then I can go for the cytokine. Cytokines. Here, what cytokines? We are having the two different cytokines are there: pro-inflammatory cytokine as well as anti-inflammatory cytokine. Here, I selected two pro-inflammatory cytokine, interleukin 1 beta, TNF alpha. Another one is another cytokine is anti-inflammatory cytokine, TGF beta. Here, before going to that, here once again 5 important steps are there, because please focus on a student, any MSc students are there. I can share this slide and any papers also. Please focus on this type of the paper. Publication is very easy. Okay, 5 important steps are there. The first one is selection of the medicinal plant. After that, extraction, phytochemical analysis. After phytochemical analysis, synthesis of the nanoparticle. After synthesis of the nanoparticle, another thing, confirmation of nanoparticle by using spectroscopic as well as microscopic analysis. And after the confirmation of nanoparticle, we can go for the preliminary screening of anti-inflammatory activity. And after the preliminary screening, we can optimize the concentration. After the optimize the concentration, we check the anti-inflammatory activity by checking the marker. And if it is showing anti-inflammatory activity, furtherly, I can check the pro-inflammatory as well as the anti-inflammatory markers and the cytokines. Okay. Then you see here, as I told you, the first step is selection of the medicinal plant as well as what? Extraction, and after that I can go for the phytochemical analysis. Here secondary metabolites are there. Very well. Primary metabolites as well as secondary metabolites. Very well. The plants does not have any immune system. Okay, only vertebrates having immune system. But how plant protect against diseases by using these secondary metabolite? Okay, you see, the plant produce diversified secondary metabolite to protect against itself against infections. Are you getting? That's why due to lack of immune system. That's why all the plant material, if they contain the secondary metabolite like phenolic compound, alkaloids, flavonoids, saponins, and tannins, carbohydrates, and all those things. These I tested by using the different test, and it is showing the presence of all the secondary metabolite, because that's why the plant containing all the secondary metabolites responsible for the pharmacological activities such as anti-inflammatory or anti-cancer or everything. That's why here another important point I didn't mention here. Already plants having its own important, but why we are not consider the plant are bulky in nature. When plants are bulky in nature, it can perform optimized biological activity. That's why I mixed these plant extract with metals. When I mixed plant extract with metals, automatically the bulk nature will be reduced. You know what is the meaning of nanotechnology? Nanotechnology, the science which deals with the fabrication of the particle in the nano size, 10 to the power of -9 to 10 to the power of -11. That's why you see in the first step, I selected the plant and I extracted and then the phytochemical analysis. In the phytochemical analysis, I identified all the secondary metabolites responsible for synthesis A, B, C. Figure 2. Here, two different figures are there. Okay, extract when I mix it and the plant extract and nano. Before going the spectrophotometer method, we can go for visualization. Okay, by using visual observation, we can confirm the formation of the nanoparticle. You see, that's why here figure 1A and figure 2A, which can confirm the change in the color due to surface plasma resonance, which confirm the formation of nanoparticle. That's why here important thing is the confirmation of the nanoparticle is the first step is visual observation. After the visual observation, I done spectroscopic analysis. You see, you may be asked, what is the relation between AA and uh, synthesis of green synthesis? Nowadays, we cannot do, no need to do everything because already so much of data is there. We no need to do because already when we can integrate this type of data with AA tools, already it can analysis it over. For example, what color of the gold is yellow color. After confirmation of nanoparticle, it can change into the brown color and whitish color precipitation. It can take the available data, and from the available data, it integrated into the it integrated our result, and it can give the result nanoparticles are formed or not formed. Okay, then the first initial observation is color change, and after initial observation of color change, when we confirm the nanoparticles are formed, we can go for the spectroscopic analysis. In the spectroscopic analysis, we can confirm the both gold as well as titanium dioxide nanoparticle by observation of their peak at 294 and 320 nanometer respectively. Clear? That's why these two different peak, for example, 294 the peak, maybe not 294, 292, 320 also there, but we got the 294 and 320 to 360 also. But this is not, I didn't give the range, I gave the optimum peak. That's why these two different peaks is responsible for the confirmation of the nanoparticle. That's why for example, my result I can confirmed with, I can check with the other existing results. That's why my results are similar with the already existed data. But here mine is 294, here is 330, but other scientists reported 315, 320 because range will be different because 60 to 70% of nanometer range will be there. That is that is a narrow range, but in the narrow range we can select our particular distant range. That's why these 2 nanometers confirms the formation of nanoparticle, both gold as well as titanium dioxide nanoparticle. Then another important thing, FTIR. You know, FTIR is the technique which is useful for identification of the functional groups responsible for the synthesis of the nanoparticle. Here you see, here AQ means aqueous extract of the plant extract, nanoparticles as well as gold nanoparticle, titanium dioxide nanoparticle. You see, when you see these three different column, aqueous extract containing alkyl group, alkane group, amide group, because the secondary metabolite, alkali, terpenoids, flavonoids, phenolic compound, everything. You see, the same extract when we prepare gold nanoparticles, some of the functional groups are disappeared. The disappearance of the functional groups, the disappearance of the functional groups itself
మ్యామ్ మీరు పంపించిన
హలో
కాదమ్మా
హలో
అయ్యానా సరే సరే ఓకే థ్యాంక్ యూ
హలో హలో ఆయన సార్ మ్యూట్లో ఉన్నారు, అన్మ్యూట్ చేయాలి. హలో సార్, అన్మ్యూట్ యువర్ మైక్ సార్. హలో హలో యా హలో హలో సార్, నాట్ ఆడిబుల్ సార్, ఇట్స్ వెరీ లో వాయిస్.
Good afternoon to all. Due to technical issues and poor connectivity, Sir is not able to connect properly and we could not hear Sir clear. So, I am proposing the concluding session for this. Thank you, Sir, Dr. Krishna Chaitanya Kari, for your enlightening presentation on innovative approaches in chemistry for sustainable solutions. Your insights into sustainable chemical innovations and their applications in healthcare and environmental science have been truly thought-provoking. As you explained your research work regarding the synthesis of nanoparticles, especially gold and titanium oxide, by using plant extracts, solanum plant extract, which could function as a usually plant-based agents will not have side effects. That point you related. So because of that, you have chosen the plant-based biosynthesis of nanomaterials. Wonderful aids in the synthesis part as well as characterization techniques and all. And you also pointed out one point of chemistry, nitric oxide, which can function as a cancer marker to assess the levels of this anti-cancer activity. So all the plant metabolites like alkaloids, terpenoids, flavonoids, all such kind of things that analysis also to some extent we cover in your presentation. And finally, this wonderful presentation has given wonderful insights about uses of AI or technology in the nanosynthesis. So finally, you adopted AI based. You made it very clear that instead of doing 3 to 4 months work manually, you can adopt suitable AI tools for the synthesis of these nanomaterials and their characterization before sending to the real world. We can get some predictions by using AI. So wonderfully, you integrated this AI technology into nanosynthesis. Thank you so much, sir, for enlightening us the uses of technology in this nanosynthesis. And now the floor is open for discussions. Hope any of our students could get to you with the proper question. Hope you are listening to us. Sir, is there one? If anyone has questions or need any clarity in the presentation given by sir, to pose a question. Sir is not there. Yeah. So we can wind up. Thank you so much, sir, for the wonderful enlightenment session.
I would like to extend my heartfelt gratitude to our distinguished speaker, Dr. Krishna Chaitanya Kari, for his explanatory, edifying, and reflective address on innovative approaches in chemistry for sustainable solutions. Your profound expertise and perspectives have certainly broadened our understanding of this specific field of study. Thank you, sir, for taking the time to share your knowledge with us. I would also like to express our sincere gratitude to our esteemed co-chairperson, Dr. Vani Lata, for her invaluable support and guidance throughout this session. Your presence and contributions have been essential in making this session a success. Thank you, Madam, for your dedication and commitment. We now proceed to oral session. It is a great pleasure to extend a warm welcome to all the participants presenting their incredible research and review papers during this session. Your contributions are of utmost importance to the success of this conference, and we are excited to hear your valuable insights. We encourage active engagement, thoughtful questions, and aim to make this session both interactive and enriching for everyone involved. I now invite Mr. K. Krishna Manohar, Head Department of Biochemistry, to present the paper entitled Nanotechnology Meets Neurodegeneration, Zwitter Ion Coated Nanopolymers for Precision Therapy. I invite you, sir.
Good afternoon, everyone. Myself, K. Krishna Manohar, Head, Assistant Professor, Department of Biochemistry, Management Technology. Today, I will deliver a talk on nanotechnology meets neurodegeneration. How the nanotechnology influences the neurodegenerative treatment. Before going to that, you have to know about what is meant by neurodegenerative diseases. So, name itself indicates neurodegenerative diseases are the diseases where neurons are progressively degenerated. So, whenever the neurons are progressively degenerated, then it leads to loss of cognitive function and motor functions. There are several neurodegenerative diseases. The most prominent ones are Alzheimer's disease, which is caused due to degeneration of dopaminergic neurons. So dopaminergic neurons are the neurons which can produce dopamine. So which is a neurotransmitter which can passes the signal between the neurons. That is nothing but postsynaptic neuron, presynaptic neuron. And these neurotransmitters are released to the synaptic cluster. So the another major prominent factor is tau. Tau is a protein which is responsible for stabilization of microtubule. Suppose if it is altered, what happens? The neurons get tangled. So if the neuron gets tangled, automatically the signal is not processed and it is not transformed from the one cell to another cell or one neuron to another neuron. So other risk factors are family history, heart diseases, and genetics. Blood-brain barrier. So it is a protective layer which surrounds the blood and the brain, and which is highly selective barrier. There are four barriers in the membrane. The first one is impermeable junctions, which will not allow any type of molecule. Permeable junctions, which will allow all types of molecules. Semi-permeable, which means which will allow some type of molecules, and selectively permeable means which will allow only specific molecules. Here, the blood-brain barrier is selective barrier, so which will only allows oxygen for the survival of cell and glucose for the nutrient, because the brain cell always craves for the glucose. Then amino acids for the production of neuroproteins. So generally, 90% of the drugs are not entered into the blood-brain barrier because the blood-brain barrier is highly selective, but also it is a barrier for the infection and also the toxic drugs. That means, so drugs are not easily entered into the blood-brain barrier and reach to the brain. So if you see the diagram, the first normal blood vessel, which contains only single layer of cells, that is nothing but endothelial cells. Then coming to the second diagram, it is a brain cell which is supported by four protective layers. The first one is nothing but astrocytes, and the second one is nothing but pericycle, and the third one is nothing but basement membrane, and fourth one is nothing but endothelial cells, which also contain tight junctions. You all know that tight junctions are the junctions which are not permeable anything. Go for the next slide. So challenges during the drug delivery to the brain. Tight junctions, you know that, which will not allow any type of molecule. Efflux pumps, they easily efflux what are the drugs which are inside the brain. And lipophilicity is nothing but hydrophobic nature, water-hatable nature. But unfortunately, 98% of the drugs we are using is hydrophilic, not hydrophobic. So that is the cause most of the drugs are not entered into the brain. So there are generally 6 types of methods, 6 types of transport systems, how the molecules are transferred into the brain. But nanoparticles use only 2 methods, that is nothing but adsorptive transcytosis and receptor-mediated transcytosis. Adsorptive transcytosis means nanoparticle having positive charge and this blood-brain barrier having negative charge. Why the blood-brain barrier having the negative charge? Because proteoglycans, endocons, perlocons. So, because of negative nature and of the positive nature of the nanoparticle, we are having electrostatic interaction between the positive and negative. Then automatically the vesicle enter into the brain cell and releases its contents. Then the next two, receptor-mediated transcytosis. in this, the ligand binds to the receptor, then receptor gets a conformation change, then ligand including receptor, that is like a ligand receptor mixture is internalized by means of vesicle and is transferred to the other side of the cell. If you see the second one, receptor mediated transcytosis, the ligand binds to the receptor, then the both are internalized, then the receptor releases the compound into the inside. That is nothing but sixth one is absorptive, positive and negative electrostatic interaction. Next slide. Right. So NDD, it is not repetition. NDD means nano drug delivery, and the second NDD means neurodegenerative therapy, neurodegenerative diseases, NDD. So generally, most common use in nanoparticles for the drug delivery is two types: deuterium coated nanoparticles and like polyethylene glycol coated nanoparticles. So deuterium coated nanoparticles. What are deuterium coated nanoparticles? What is meant by deuterium? Nothing but a compound which is having both positive and negative charges. What is meant by deuterium coated nanoparticle? If the nanoparticle is coated with deuterium, then it is considered as deuterium coated nanoparticle. So scientists already prepared a artificially synthesized deuterium particle that is called as PMPC, and which is also having an additional property which structure resembles the one phospholipid in our body that is called as phosphorylcholine. That means having two additional features. The first one is deuterium, that is hydrophobic, and the second one is nothing but which also contains a structure which is similar to phospholipid structure. And the second, PEG-coated nanoparticles. So PEG-coated nanoparticles are hydrophilic in nature. So whenever they are coated, I already told that blood-brain barrier is lipophilic, that means hydrophobic. So here there are two nanoparticles. One is deuterium, that is nothing but hydrophobic, and PEG-coated nanoparticle is hydrophilic. I already told that blood-brain barrier is hydrophobic. Which molecule will enter? Simply a deuterium-coated nanoparticle. Because it is hydrophobic, the blood-brain barrier is also hydrophobic. Coming to next slide. Then how to synthesize the nanoparticle and how to characterize and how to check the efficiency. Already Krishna Chaitanya sir told that there are several methods to synthesize the nanoparticles. These are the other method. The first one is ring open polymerization. In this process, we will convert the cyclic compound into a simple compound, that is a linear molecule. Then next we will go for flash nanoprecipitation. By these two methods, we will synthesize the nanoparticles. Then go for the next slide. So after synthesis, we will check the purification by using cellulose microfibers or cellulose material. Then after that, then we will go for characterization by means of chem and SEM in order to check the size and its other morphology and other things. Then directly we will go for animal studies. Before going to animal studies, first we will check on the cell culture lines. So we are taking two cell culture lines here. BEN3, BEND3 is a mouse brain cell line because the blood-brain barrier is present in brain. That's why we are taking the brain cells. So BEN3 is a mouse brain cell line. Then automatically N2A is a neuroglial cell line. So for the both cells, we are injecting nanoparticles. First one is PEG-coated nanoparticle, and the second one is PMPC-coated nanoparticle. Go for the next slide. Go for the results. That is the graph. It is actually the kinetics portion. So in the x-axis, you are taking the nanoparticle concentration. In the y-axis, you will go for intensity, nothing but a fluorescence. How many molecules are internalized? So in the first thing, PEG-coated nanoparticle, after 24 hours, is showing absorbance at around 8,000 to 10,000, right? Then coming to the PMPC-coated nanoparticle, showing absorbance after 24 hours around 60,000. It clearly indicates that PMPC-coated nanoparticles are more internalized than compared to PEG-coated nanoparticles. Suppose if we consider the confocal images, the first one is control, we are not treated with PEG or PMPC. Why it is in green color? Because of the neuron cells which are tagged with the green cell protein and nucleus which is tagged with the highest state. Then PEG-coated nanoparticles, after 4 hours, a slight internalization is seen. After 24 hours, a little bit increased. Go for the PMPC, after 24 hours, you see the red color. That means more number of internalization. the more nanoparticles are internalized. Then coming to this is about mouse brain cell line. That means we confirm that in the mouse cell line, the PMPC coated nanoparticles are entered or internalized into the brain. Then coming to the next one, N2A, that is nothing but neuroglial stem cells. Sorry, neuroglial cell line. So in this, the first one is control. The first thing is a black and white color, which explains about the cell morphology and cell structure. You go for the PMPC coated nanoparticles, there is a white cell, square shaped box there. There the cells are aggregated. Then coming to the internalization, then you easily, after 24 hours, PMPC showing the more internalization of nanoparticles. That means cell lines we are completed. Then for directly we will go for animal studies. The animal we are choosing is zebrafish because it is having 70% similarity, and also the blood-brain barrier of humans is exactly correlated to zebrafish because it contains FLX forms, it contains proteome life forms, and everything. So that we are recommended zebrafish as a recommended model, animal model. Go for the next slide. So before going to that, we have to check if suppose the nanoparticle is interacted with the biomolecule, what is its character? Then we will go for protein bound assay. For the protein bound assay, we are actually taking a protein called as albumin. Why we are only taking albumin? We already know that there are two mechanisms nanoparticles are internalized: adsorptive transcytosis and also the receptor mediated transcytosis. Adsorptive transcytosis means nothing but an interaction between a positive and a negative charge. Albumin always having the positive charge, so that's why we are taking the albumin and we are tagged with a fluorescent protein. So fluorescent albumin, and this is tagged with nanoparticle. Then in order to check whether the nanoparticle uh, modifies any protein structure or not, or whether it is a neutral one. Then coming to the animal studies, the zebrafish larva. So confocal microscopy, exactly you go for fifth picture. So if you compare the second picture and fifth picture, in the second picture there is internalization, but a little bit. When compared to the fifth picture, the internalization is more. That is nothing but is the trunk region. Trunk region nothing but head region. So trunk region of the zebrafish showing high amount of nanoparticle internalization. It clearly indicates that PMPC coated nanoparticles are easily internalized. Deuterium coated nanoparticles are easily internalized compared to the PEG coated nanoparticles. Next slide. So, deuterium nanoparticles shows a promising drug delivery strategies in neurological conditions, exceptionally to treat the neurodegenerative diseases like Alzheimer's and Parkinson's. Thank you.
Presentation. Dear students, scholars, faculty, I kindly ask you to proceed to the multi-purpose hall in the first floor of this block where lunch will be served. We request the student volunteers to assist the dignitaries and attendees in reaching the venue. We encourage and respectfully ask everyone to return to this conference hall by 1:30 p.m. for the continuation of the proceedings. Thank you. Professor at the College of Pharmacy, KL University, Vijayawada, Andhra Pradesh, to the stage. Sir, may I request you to kindly come on to the dais. VP of Microbiology will welcome Sir with their sapling. Thank you, sir. Thank you, VP. It is now my privilege to invite Dr. P.V. Rekha Devi to the dais as the co-chairperson for this session. I request Shafia of Microbiology to welcome Madam with their sapling. It is now my honor to invite our co-chairperson, Dr. P.V. Rekha Devi, to introduce our esteemed invited speaker, Dr. Kashi Vishwanath. Please, ma'am. Good afternoon, everyone. It is with immense pleasure that I welcome Dr. Kashi Vishwanath sir amongst us for this today international conference. I take this privilege in introducing Sir to you all. Dr. Kashi Vishwanath is presently working as an associate professor in KL College of Pharmacy, KL Educational Foundation. Dr. Kashi Vishwanath completed his M Pharmacy with pharmaceutic specialization in 2011 and worked at Annamacharya College of Pharmacy affiliated to JNTU Anandamam. Later, he worked as an assistant professor at Srivasavi Institute of Pharmaceutical Sciences, Tadepalligudam for about 4 years and then joined as a full-time research scholar and teaching assistant at the Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, DHU Varanasi in 2016. Post submission of his thesis in 2021, he got awarded with PhD in the year 2022. Later, Sir worked as an assistant professor at Geetham University, Hyderabad, and Sir is currently working as an associate professor at KL College of Pharmacy. In the span of over 12 years of teaching and research experience, Sir has published about 35 research articles and review articles and 4 book chapters in peer-reviewed, high-impacted international journals. He has almost 1,000 citations with an H index of 18 and an I index of 23. His doctoral research focused on the design, development, characterization, and evaluation of biocompatible EGFR targeted cytosine nanomedicine and redox sensitive nanomedicine for the treatment of non small cell lung cancer. For this, Sir developed a GSH redox sensitive TPGS SH with high affinity towards cancer microenvironment. He also worked on the development of targeted nanomedicine for breast cancer, tuberculosis, and glioma. He has gained comprehensive knowledge about various methods of optimization and preparation of nanoparticles such as liposomes, missiles, polymeric nanoparticles, upconversion nanoparticles, and biodots, etc., and techniques involved in the characterization of nanosystems both in vitro and in vivo. Currently, Sir is involved in the formulation of natural inspired nanofibers for the improved wound healing applications, hydrogen based microparticles, mixed missiles for the treatment of various cancers. Sir is currently guiding about 5 PhD scholars and 4 PG students. With this, I welcome and extend my hearty welcome to Sir on the occasion of our international conference, and I now hand over the stage to Sir for his inexpressible.
Thank you, madam. Thank you for the introduction of this. As you understood from the introduction, I put up my experience on analysis of development of cancer. And I know it's a big experience, so I try to make this presentation and then during this start of session. Now, this is on the development of diagnostic systems for simultaneous diagnosis and therapy.
I request Shafia of microbiology to add a little
So before going into the actual session, so I want to just tell you about the persons who are the development of identity. Pandit Madan Mohan Malviya, who was the founder of PHU, IIT PHU Varanasi. And that university is inaugurated, established before 100 years, 2016, yes. 1916, and so almost I think 2019 we celebrated centenary celebrations, 100-year celebration of the university, and the university's span so went across I think 4,100 kilometers, 100 acres of campus is there in Varanasi, and all the departments exist in the university. So I want all of you to target to aim for admission into such kind of universities where you can do very excellent research, where you can grow yourself as well as you can contribute to the development. So not only BHU, we have the Institute of National Importance, primary institutes. Because you are biotechnology students, you have bright opportunities in the field of research, particularly medicinal research and the development of these drug delivery systems and other things. So I request all of you to target to get this admission to one of such universities. And I myself was fortunate to get admission in that university for my PhD. Yes. Is actually before coming here, I thought this is a pharmacy department, but after coming here, I came to know that pharmacy is not here. So, Dr. Mahadev Lal Shroff, who established the Department of Pharmacy in BHU, and he introduced the B Pharmacy and M Pharmacy programs in the BHU. So he's named as the father of pharmacy education in India. I want to give you a brief glimpse about him. He developed so many things. He worked in so many institutes, India and abroad, China, Japan, and some other places. And after coming back to India, he joined in Birla Institute. Then he came to BHU, and he started the Department of Pharmacy there. Then he contributed to so many milestones in the development of professional pharmacy. So coming to our topic, cancer. I think all of you know, all of you well aware of this cancer. So what is cancer? It's a uncontrolled growth of cells. So generally there exists a tight control over the division of cells, growth of cells. But what happened? Cancer escape all the regulatory control and it grows in whatever way it wants. And in India, so there is a concern about growing number of cancer incidences. Almost 14 lakhs of reported cancer cases every year, and almost 6 to 8 lakh of people are dying of cancer every year. Next. So this is how cells develop into cancer. So of normal cells, their division and everything is tightly controlled, and there exists a tight borders for the development of tissue. But where we are coming to cancer, there is no control. So cells will develop as they want, as they like, and there is no control over the shape of the cell as well as the size of the nucleus, and they escape the regulated borders. And their chromosomes also, they are not regulated. They grow like whatever they want, so there is no regulatory control over the growth of cells and division of cells. So what I told you just, so we have growing concern about the cancer incidences in India. So almost 14 lakh cancer cases were reported every year, and this number is rising alarmingly. And there are different types of cancers, I think. So the main cancer affecting women is breast cancer, and other types of cancers like lung cancer, mainly cause what is the cause of lung cancer? I think tobacco smoking. So there are almost 60 different compounds, established compounds, which will cause cancer. That they cancer causing activity that is called as carcinogenicity. That is fully established. So 60 different compounds are there in tobacco smoke. So unfortunate thing is most of these cancers are preventable. Cancers caused by tobacco are preventable, but people are not following those instructions. And then next step, most prevalent cancer is colorectal cancer in both men and women. So coming to the main aim behind this session is the nanotheranostics. So theranostic actually is a combination of two words, therapeutics as well as diagnostics. So mainly in the treatment of cancers, what's the problem is diagnosis play a key role because we have to continuously monitor about the therapy, the progress of therapy. Once you give the drug, you have to monitor the therapy progress and how the cancer cells are dying. We have to monitor. So that becomes the major issue. So now this diagnostic platform will provide the opportunity to include both therapeutic, that means drug as well as diagnostic agent. So you know how the diagnosis is performed generally. We have various diagnostic techniques like X-ray, CT scan. So for that purpose also generally if you see if the doctors want to diagnose heart or some internal tissues, they give some contrast agents. You have heard or not? So like barium fluid, for example, if you are going for X-ray of GIT, doctors will give you initially before going to scan, they will give you barium fluid, and that fluid will obstruct the X-rays so that wherever this fluid is present, doctor will get the image of the particular tissue on the X-ray film. So these diagnostic agents require some radiation to get to show the signal. So now, this diagnostic platform will include both diagnostic agent as well as the drug. And when it is given to the patient, it reaches the cancer cell. And once it reaches the cancer cell, some type of radiation is passed over the cancer area so that that diagnostic agent will emit some fluorescence or some radiation that will be captured. So that's how we can monitor the progress of the therapy. So that is the main aim behind the diagnostics. As well as so how to know progress of the therapy in internal tumors. This is the main question behind this concept, development of concept diagnostics. So as I discussed, the nanotheranostics is a combination of imaging and therapy in a single nano platform, and it can be used to diagnose as well as treat diseases like cancer, HIV, and tumors and other diseases also. And recently, our lab in BHU they developed one system to monitor the blood clotting. What do you say? Embolism, thromboembolism. So they developed a system which actually go to the particular site where the blood clot happens in the internal blood vessels, and we actually get an image of the blood clot. So nanotheranostics provide best advantage. So simultaneous treatment as well as diagnosis, with which we can revolutionize the cancer therapy. And personalized medicine. Do you know what is personalized medicine? Till now we know about medicine, but we don't know about personalized medicine. That's a new concept. So based on the genetics, treatment will be provided at individual level. So each and every individual is different in terms of genetics. So if you know about the genetics of the person, we can give the treatment which exactly suit to the particular person based on the genetic buildup. So that is personalized medicine. So this diagnostic also helpful to design a personalized medicine for a particular person because every individual is different. Every individual responds differently to the treatment. So that's why a new concept is evolving, that is personalized medicine. So this is the diagrammatic representation about the diagnostic. So we load both drug as well as diagnostic agent in single nano platform, and when it is given to the patient, it is the cancer cell, and simultaneously, if you pass some radiation into that area, the person or the operator will get the signal about the cancer. And you may have a doubt how we make the nanosystem or nanoparticle to exactly reach the cancer cell. Any idea about that? So we have to use some targeted moieties, some antibodies, some proteins, some aptamers, which make this particle exactly reach the the cancer cell. Because cancer cell overexpress certain things which are not present in the normal cell. So we target some aspect of the cancer environment of cancer cell, so that one of the particle we design that will make it reach to the cancer cell, not to the normal cell. And these are the some of the components of the thermostat system. So the left, extreme left at the top, this is gold nanoparticles, neuropods, quantum dots, iron oxide nanoparticles. So these are the diagnostic agents which generally loaded into the nanoparticle. Along with that, we can load different drugs, either chemical drugs or biological drugs, proteins, antibodies, etc. And these are all these together loaded into some polymeric nanoparticles or some other types of nanoparticles like liposomes, etc. So these are the some of the diagnostic techniques which we use to monitor the therapy using this diagnostic platform, like MRI. What is MRI? What is so magnetic resonance imaging, okay? Computer tomography, positron emission tomography, okay? Optical imaging. So optical imaging, what we do, we use some fluorescent dyes which are loaded into particle or any third system. So when you pass some radiation, that will produce some signal. That is optical imaging. And then single photon emission computer tomography, that is spectrum. So these are the some of the diagnostic techniques which are used to track the therapy in case of cancers. So to monitor or to image the cancer site, all these techniques require some carrier or some system in which you load the drug along with the diagnostic agent. So in all these types of techniques, we require some carrier system. Scrambled. So these are some of the examples. Some of the examples of diagnostic agents which are loaded with drugs and with the nanosystem. For example, you see here the iron oxide, the mRNA, and mixed. Next. Till now we discussed that nanoparticle is like a very good, very excellent. But the problem is what happened? Any foreign particle when introduced into the body, either orally or when you give injection into the body, blood circulation, what happens? Because it is a nanoparticle is also a foreign particle. What happens? It quickly recognized by our immune system, so opsonization, and then it leads to phagocytosis. So if you make the surface of the nanoparticle hydrophilic means water soluble. So if you make the surface of the particle hydrophilic, what happens? It can escape. There are more chances that it escape the uptake by phagocytosis or immune competencies. So this is what the concept behind this. So if you want to make the surface hydrophilic, we attach some hydrophilic moieties like polyethylene glycol, which is known as pegylation. So by giving pegylation, so what we do, we can make the nanoparticle to stay more time in the blood circulation by escaping the uptake by RES or immune cells, so that it can actually reach the target site and produce the effect. So that is what the main objective of pegylation. And another thing is we can use a surfactant known as TPGS. So actually, TPGS is a combination of 4 different chemicals. One is vitamin E, and then PEG, polyethylene glycol, and one succinate molecule. So these 3 are there in the TPGS. So T alpha tocopherol. Tocopherol is vitamin E, and polyethylene glycol 1000 succinate. That is the full name. So this TPGS is very versatile. So it has so many applications. And one of the important applications is it is a PGP inhibitor. So you know what is the role of PGP? PGP is a efflux pump. Whatever the drug that reaches intracellularly, this pump exists on the surface of the cell and throw the drug out of the cell. This is also there for the protection of our cells, but it will throw the drugs also out of the cell. So TPGS can inhibit the PGP pump, which will enhance the effect of anti-cancer drugs, because PGP pump is the main reason behind the development of resistance to most of the anti-cancer drugs. So if you can inhibit PGP pump, what happens? The efficacy of your therapy can be improved. So that is why. So TPGS is maybe included as an important component of the nanosystem for many of the advantages. It is also helpful in the emulsification for the preparation of nanoparticles, and it is a solubilizer, it is a stabilizer. So many applications are there. Along with that, the important application is PGP inhibition. And so we can load different types of drugs, either solid drugs or solutions or liquids or proteins, antibodies into nanotherapy system. So the applications are unlimited. Now we'll see one of the recent work which has been done in this same lab, our lab, that was in nanotherapy. So, the breast cancer is mainly affecting the highly influencing the women. So, highly incident cancer in the women, and almost 2.3 million cases are reported every year in the US, and around 60 to 70% people are dying out of these reported cancers. 60 to 70% of people are dying from this cancer. And we have different two types of cancer. One is hormonal type of cancer, and one is triple-A2 breast cancer. So hormonal means this cancer produces hormone receptors like estrogen receptor and progesterone receptor along with HER2 receptor. The HER2 receptor is one of the receptor which is helpful in the cancer growth. Generally, it is helpful for the generation of new blood cells and other things, but cancer uses it in a derogatory way. So that's why. And other type of cancer is triple-A2 breast cancer, which is very difficult to treat because it has no receptor, so you cannot. It is very difficult to target this type of cancers. And so for this type of cancer, so our lab developed palbociclib loaded fluorescent active nanocluster reduction system for the treatment of breast cancer. So we'll see how it is developed and what are the different evaluation parameters about this. So this is the hypothesis. So what they did actually, they induced the breast cancer by injecting some chemical. I told you one of the chemical from cigarette smoke, DMBA. So dimethyl bench anthracene, DMBA, which is a very known chemical, very established chemical for the induction of breast cancer. Just you inject this chemical into the breast pad of the rat, it will induce the cancer within days, 1 or 2 days. So that chemical is used to induce the cancer in rats. And what was the system prepared? So it contains different moieties. One is FNC. FNC is nothing but fluorescent nanoclusters. That is the diagnostic agent. And PBB is the drug, palbociclib. CS is chitosan or chitosan, which is the polymer to make the nanoparticle. And SS stands for disulfide linkage. Actually, there is a reason for including this disulfide linkage in this particle. What it will do, when the nanoparticle reaches the cancer cell, so cancer cell has some peculiar qualities as compared to normal cell. The pH is very low, 3 to 5, and the GSH concentration. What is GSH? GSH is nothing but glutathione. So glutathione is present throughout our body normally in low concentration. That is 1 to 5 millimolar concentration is there in normal cells. But in cancer cell, the concentration will increase up to 20 millimolar. Because cancer cell want to escape from all the immune attacks, so it increases the redox potential to 20 millimolar. It produces high quantities of GSH. So this particular disulfide linkage which we include in the nanosystem is sensitive to high concentration of GSH. That means as the particle reaches the cancer site, the high concentration of GSH will cleave the bond which is present in the particle so that the particle will release the drug. That is the hypothesis. Understood? So one is the diagnostic component FNC, palbociclib is the anti-cancer drug, and chitosan is the polymer, and disulfide linkages are there to make the drug exactly release at the cancer site. We made the nanoparticle. So when it is given to the cancer cell, because of the high concentration of GSH, that triggers the partial release of the drug at the cancer. So this is the procedure for preparation of nanoclusters. So very simple. We use calcium chloride, bovine serum albumin, and ammonia, sodium hydroxide. Only 4 chemicals are used to produce these nanoclusters, fluorescent nanoclusters. So if you see, if you see the nanoclusters producing the fluorescence, if you excite those nanoclusters at the wavelength of 366 nanometers, it produces blue fluorescence. And when the same nanoclusters are excited at 469 nanometers, they produce green fluorescence. So this fluorescence will guide the therapy. You can actually get the image of the cancer tissue. And this is the brief procedure for the preparation of nanoparticles. So just we have to first prepare the fluorescent nanocrystals, and then that is added to chitosan solution along with the drug, and it is probe sonicated, and further the nanoparticles are prepared. So the nanoparticles are separated from the suspension by centrifugation at 20,000 rpm. And once they are separated, they are resuspended into some distilled water and stored for further. These are the some 10 images of nanoparticles. You can see the scale is 200 nanometers. The size, maybe the size is around 100 to 150 nanometers only, the prepared particle. These are the some of the in vitro evaluation parameters of prepared nanoparticles. This is the XRD evaluation. You know what is the advantage or objective of the XRD evaluation? What we use XRD for? So XRD, X-ray diffraction, we use to study the crystalline pattern of the drug. So when before loading of the drug into nanoparticle, drug is in crystalline form, but after loading the drug into nanoparticle, the drug will convert to amorphous form. So we know the crystalline form, if you compare the solubility of crystalline form to amorphous form is more solid. So to prove that, we did this X-ray diffraction analysis. You can see here. Solid. And those are the in vitro drug release studies at the different pH. One is at 5.5, one is at 7.5. As I already told you that cancer cell has lower pH, around 3 to 5. And as the drug reaches the cancer cell, the drug will be released in higher quantity. So that to prove that, we conducted the in vitro drug release studies at different pH. And the particles showed higher drug release at lower pH of 5.5. That proves that these particles are useful for cancer therapy. And these are the hemocompatibility studies. So these studies are conducted to prove that what are the system we prepare that is safe to the blood cells. So what we do here, just we incubate the prepared nanoparticles with some 2 ml of blood, and after brief incubation, those samples were observed under microscope after staining with Hellman reagent. So once you see this is the clean water. Other two are formulations. One is redox phase 2 and another one is non-redox phase 2. That means one particle contains SS double bond, other type of particles do not contain SS double bond, disulfide. So apart from the pure drug in water, apart from the pure water, all other systems are proved to be safe to the blood cells. They do not cause any harm to the blood cells, RBC. So whatever the system, nanosystem we prepared, that is safe for the blood cells. What happens when the blood is treated with pure water? You see, hemolysis. Why? I'm asking like I want to make you more. So because you know, if you inject pure water, what happens? Because of the osmosis, blood cells will undergo lysis. That's the problem. So, but our nanoparticles are proved to be safe to the. These are the cellular uptake studies. So here, what we do, the fluorescent nanoclusters loaded nanoparticles are treated to the cells, MCF7 breast cancer cell line in lab, and after treatment of the cells with these particles, they are stained with propylium iodide. So propylium iodide, what is the main objective of treating with propylium iodide dye? So these propylium iodide dye will stain the dead cells. It can enter into the cells which are dead only. Live cells cannot uptake the propylium iodide dye. So to prove that these nanoparticles are more uptaken by the cells, we treated with first nanoparticles and then stained with propylium iodide. You can see here. This established that whatever the nanoparticles prepared with disulfide bonds are taken by the cells in huge numbers as compared to those particles which do not contain disulfide bond linkage. These are the cellular apoptosis study. So here what we did again, we want to prove that our particles are actually killing the cancer cells. So for that purpose, we treated the cells with different formulations again. Only for control. Control is just treated with normal saline and this PBB pure drug in water. That is another treatment. And two types of nanoparticles. One contains disulfide bonds, another do not contain disulfide. So 4 treatments we have done. And you can see that. That's why it is proved that prepared nanoformulations are more cytotoxic to the cancers. And this is the main study, that is in vivo anti-cancer study. So actually, as I told you initially, so we use the cancer in rats by injecting DMDA, that is the chemical from cigarette smoke. And after the treatment with this agent, every 2 weeks, the formulations are injected into rats, and the size of the tumor is checked every time and so. Now you see, so 3rd day, 6th day, 9th day. So control is only normal saline without any drug. PBB is pure drug. And 2 different types of formulations are given to the 4 different groups of rats which are induced with breast cancer. And you can see compared to all other treatments, these nanoparticles containing disulfide bonds will completely cure the cancer. The cancer tissues almost disappear. So you can see the control, the cancer tissue size is maintained as it is. The pure drug also only insignificant effect on the cancer death. Our both formulations they shown better effect, but between these 2 formulations, the 4th formulation actually completely eradicate the cancer tissue. So this established that these prepared nano formulations are actually curing the breast cancer, completely eliminating the cancer cells. And the graph shows the percentage of cell death in the cancer tissue, comparison between 4 different treatments. And this is the in vivo imaging. So we want to prove that what are the system we prepared that actually reaches the cancer site or not. Because whatever you inject into the blood circulation, that will be circulated throughout the body. And we want to prove that these particles are exclusively reaching the cancer cells. For that purpose, the fluorescent nanocrystal loaded nanoparticles are injected to rats, and then they are imaged with in vivo imaging system. So here you can see that this red signal actually related to the fluorescent nanoclusters. So from the targeted nanoparticles will be prepared with disulfide linkage, they reach the cancer cell. So here, 4th formulation or 4th treatment reaches the cancer cell in huge number and actually produces the anti-cancer. So this is done in the in vivo imaging system. So these are the conclusions. So redox issue, cytosine nanoparticles are fabricated by solid evaporation method. Particle size, PDA is polydispersity index, zeta potential. These are the important parameters to prove the stability of nanoparticles. And these are found to be in optimal range. And the sensitized nanoparticles were tested for various physicochemical parameters, and they prove that prepared systems are stable. Particles containing disulfide linkage They displayed significant drug release at higher TS concentration at acidic pH. So what we proved in vitro drug dissolution studies at pH 5.5, particles showed higher drug release. Almost 95 to 97% of drug was released at pH 5.5, but whereas at pH 7.4, only 50 to 60% of drug released in the given time of 72 hours. And in vitro cellular uptake studies revealed enhanced uptake of disulfide link containing nanoparticles than free fluorescent nanocrystals or other type of nanoparticles. So we already proved that these fourth type of formulation, that is formulation containing disulfide linkage, they are taken by the cells in huge numbers than other treatments. And prepared formations, both the formations shown better cytotoxic activity against cancer cells, but comparing between two formations, the second formation which contains the disulfide linkage produces superior cytotoxic effect. That is established by lower IC50 value. And ultrasound imaging also proved that the cancer tissue is almost completely disappeared in case of the second formation, and the first formation also produces significant anti-cancer effect as compared to control as well as pure palbociclib. And in vivo imaging also proved that prepared formulation reaches the actual cancer site in huge number and produces strong signal, strong red color fluorescence as compared to other treatments. And this is the publication resulted from this work, which is published in Applied ACS Applied Nanomaterials. So when you do the recently, like 1 month back, this is published. That is what I want to discuss with you. I don't know how many of you actually understood what I discussed. So as I already told you, I thought that the pharmacy department. So any questions? How many of you understood actually? That is my question. That's why I wish all of you to try get admission into one of the premier institutes after completing course here. Try to pursue your research because you are biotechnology. You have vast number of opportunities post-COVID situation. I wish you all a better career and success. Thank you.
So in the chemotherapy, we are observing there are some sort of side effects as well. How can we be assured that from your treatment?
I don't say this treatment do not have side effects, but what we propose that because of the presence of disulfide linkage, so this particle exactly reaches the cancer cells, and because of the high GSH concentration, glutathione concentration, and lower pH at the cancer cell, this particle will release most of its payload, that is drug at the cancer cell compared to normal cells. That's how we propose. Actually, we did not carry any in vivo studies in humans, only studies done in animals. We have to establish it to do that. Comparing to pure drug, the side effects produced by this system is comparatively less.
Thank you.
I would like to extend our heartfelt gratitude to our esteemed invited speaker, Dr. Kashi Vishwanath, for Sir's introspectively illuminating and reflexively challenging address on applications of nanothermostics for advanced diagnosis and therapy of cancers. Your specialized skills and visionary reflections enriched our understanding of this crucial topic, sir. Thank you, sir, for your insightful talk on the applications of nanothermostics in targeted drug delivery, molecular imaging, and precision personalized medicine. Thank you, sir, once again, and your deep dive into how nanoparticles can be leveraged for biomarker-based therapies and improving therapeutic efficacy in the tumor microenvironment has provided us with valuable insights into the future of personalized medicine. Your exploration of fluorescent calcium nanoclusters for advanced imaging in breast cancer therapy is highly transformative. Thank you, sir, for taking the time to share your knowledge with us. I now invite Dr. R. Gangaraju for coming forward and felicitating Sir. Sir, kindly please come forward. Thank you so much, sir. We now move forward to oral session 3. We are delighted to welcome all the participants who will be presenting their research and review papers during this session. Your contributions are exceedingly valuable to the success of this conference, and we look forward to hearing your insightful presentations. We encourage you to engage with the speakers, ask questions, and make this session interactive and enriching for everyone involved. Without further delay, let us begin the proceedings of oral session 3. I wish all the presenters the best of luck. I would like to invite Ms. Chrysolite to present a paper entitled In Vitro Regeneration Studies on Hemidesmus indicus, a medicinally important plant using nodal explant virtually. Dear presenters, kindly make sure that your presentation is within 10 minutes. Thank you.
Hello.
Hello.
Kindly please proceed.
Hello.
Yeah, hello.
Kindly please proceed.
Hello. Chrysolite అండి. Chrysolite.
Kindly please proceed further.
హలో.
మేడం మీరు ప్రజెంట్ చేయండి.
చేస్తున్నాను, నేను షేర్ చేశాను.
Hello. You can proceed, ma'am.
Okay. So slide is visible now.
Yes.
Yeah, yes, ma'am.
Okay. So slide is changing.
Yes, yes, ma'am.
Okay. A very good afternoon to you all. Thank you for giving me this opportunity. I myself, Chrysolite, a research scholar from Department of Biochemistry, Andhra University. I would like to share my entitled just in vitro regeneration studies on Hemidesmus indicus, a medicinally important plant using nodal explants. Coming to the classification, it belongs to the Plantae kingdom and Trachyphyta phylum, Mangalypsida class, and Gentialis order, and Acyclopidae family, and Hemidesmus genus and Indica species. Its common name is Indian Sarsaparilla. Coming to introduction, Hemidesmus indicus, commonly known as Indian Sarsaparilla, is an aromatic medicinal twining shrub distributed in moist areas of India and Sri Lanka and prescribed against inflammation, diarrhea, respiratory diseases, skin diseases, loss of appetite, and epileptic fits in children. The root extract of this plant has been widely used in the traditional systems of medicine in India, such as Ayurveda, Siddha, and Unani, mainly as blood purifier, diuretic, anti-rheumatic, anti-cancer, anti-diarrheal, and as anti-viper venom. It is also used to treat nutritional disorders, syphilis, fever, foul odor from the body, bronchitis, piles, rat bite poisoning, epileptic fits in children, and Tridosha disease of blood, leukorrhea, kapha, and vata. It is also used as a flavoring agent for the preparation of soft drinks and bakery products, and also used as perfumery in cosmetic industry. Hemidesmus indicus assists in maintaining optimal liver health, anti-inflammatory and antimicrobial properties, alleviate psoriasis and eczema, and it also has carminative properties, reducing flatulence and easing the digestive spasms. Immunomodulatory properties help strengthening the immune response. The major chemical constituents responsible for its medicinal properties are luteol, vanillin, rutin, 2-hydroxyl 4-methoxybenzoaldehyde. Many of the compounds are difficult to synthesize chemically and hence they are still being isolated from the plants. In view of its high medicinal value, the plant is indiscriminately collected from natural habitat and is now rare species and getting endangered. According to National Medicinal Plants Board, it has reported Hemidesmus indicus as one of the priority species because its annual requirement is 500 to 1000 metric tons. Well identified endangered medicinal plant Hemidesmus indicus research endeavored for establishing tissue culture protocol towards their conservation. Aims and objectives. Phytochemical studies in root and leaf extracts of Hemidesmus indicus and induction of callus from leaf and nodal explants of Hemidesmus indicus. And coming to phytochemical evaluation, the preliminary phytochemical evaluation was done for the detection of alkaloids. We have gone through Wagner test and Dragon-Duff test. And coming to saponins detection, the froth test and foam test were performed. And phenol detection, ferric chloride tests were performed. And for the detection of tannins, gelatin test and detection were employed. And for the detection of flavonoids, alkaline reagent test and lead acetate test were performed. The quantitative methods for analysis of phytochemicals are performed, and the results are enclosed in the next slides. And the materials and methods. Collection of the plant material. The young healthy twigs from 2-year-old plant were collected surrounding areas of Andhra University area, and they are washed with running tap water and detergents and final rinse with distilled water. And for the callus cultures, we took leaf and nodal explants. For prior to the inoculation, the surface sterilization is needed because it's very prone to contamination. So fresh young leaves were collected from field grown plants and brought to the laboratory. Surface sterilization were carried out by washing leaves thoroughly with running tap water and with between 20 for 10 minutes and followed by the Bavastine treatment and a commercial fungicide for 5 minutes. Then the leaves were subsequently surface sterilized with 0.1% mercury chloride for 2 to 5 minutes and thoroughly washed with sterile distilled water. And the nodal segments are also subsequently sterilized as mentioned above. And for preparation and sterilization of culture media, we are using MS medium, Murashig and Skoog media, which is supplemented with different concentrations of plant growth regulators along with the combination of BAP, NAA, IAA, and 2,4-D. The pH of media is adjusted to 5.7 before autoclaving, and all the utilities were autoclaved. And the sterilized medium was poured into culture tubes and baby jars. And in the laminar flow unit, we are inoculating the media. Coming to the results, the phytochemical screening showed that the plant contains alkaloids, steroids, saponins, terpenoids, tannins, flavonoids, and phenols. Um, Hemidesmus indicus leaf extract showed positive for all the alkaloids, steroids, saponins, terpenoids, tannins, and flavonoids and phenols. As usual, the root extract also gave positive for all these. And these are the quantitative amounts of the phytochemical analysis from both the plant and the leaf extracts of Hemidesmus indicus. Coming to inoculation of explants, surface sterilized leaves were dried between Wattman #1 filter papers. Then leaves were cut using cork borer, and the leaf discs were inoculated onto the MS basal medium and also on the MS medium supplemented with different concentrations of plant growth regulators like 2,4-dichlorophenoxy, N-acetyl acetic acid, BAP, and all these, all the four concentrations. And subculturing was done after 21 days. The results were recorded after 4 weeks. For each experiment, leaf discs and nodal cuttings were used, and this experiment was repeated thrice. Coming to nodal explants, surface sterilized nodal segments dried between the Wattman #1 filter papers and were inoculated on MS basal media alone, and also MS media supplemented with different concentrations of growth regulators, BAP, NAA, and BAP. In this, I have taken this BAP with 1 mg per liter concentration and 2 mg per liter concentration, and along with BAP with 2,4-dichlorophenoxyacetic acid, which concentration is 0.1 mg per liter. Subculturing was done after 21 days. We observed a healthy and green callus on the media. The results were recorded of 4 to 6 weeks. For each experiment, nearly 120 to 130 explants were used. The experiment was repeated. And for induction of the callus, the MS medium was supplemented with various concentration of growth regulators. After culturing on MS media, supplemented with various combinations of plant growth regulators like MS media plus single concentration of BAP, and MS media plus BAP and NAA. MS media along with double concentration of BAP and 2,4-dichlorophenoxyacetic acid. The leaf explant dedifferentiated and began to proliferate the callus within 8 to 10 days. With these diverse degrees of callusing response. Although the type of callus that develops in tissue culture depends on explant and the species, it can be modified by adjusting the medium's content and the combination of different growth regulators such as NAA, IAA, 2,4-D, BAP, and all. In response to plant growth regulators, Hemidesmus indicus leaf explants produced both compact and friable callus. And it also originated the multiple shoot induction and rooting. Multiple shoot nearly 2 to 4. I observed that 3 multiple shoot generated from nodal segments on MS and BAP, which is 2 mg per liter, and 2,4-D with 0.2 concentration after 4 weeks. A decrease in number of shoots may have resulted. due to profuse callousing at base, and similar result is reported in hemidesmus indicus as of all others. And in present study, combination of BAP and NA was very effective for generating the multiple shoots. In this, the first picture shows that leaf and nodal explants inoculated on MS media supplemented with 1 mg concentration of BAP and 0.1 mg concentration of NAA. And these give rise to callus response observing at the ends of the cuttings on the leaf. So after 3 weeks, we observed the callus on MS plus BAP media. And the third picture shows that rooting response was observed from nodal explants inoculated on MS media supplemented with 1 mg per liter BAP and 0.2 mg concentration of 2,4-D after 3 weeks. Shoot formation nodal explants inoculated on MS media supplemented with 1 mg per liter. These 4 and 5 indicates the BAP and IA and NAA. And the last picture shows that multiple shoots that were observed after 4 weeks. That this arised in concentration of 2 mg per liter BAP and 0.2 concentration of 2,4-D. And coming to conclusion, from the present study, it was concluded that this plant is rich in phytochemicals such as alkaloids, saponins, tannins, flavonoids, and phenols. Callus induction was observed in leaf disc explants inoculated on MS media containing different concentrations of IAA, NAA, BAP, and 2,4-D. The best media for callus culture is 1 mg per liter BAP and 0.1 mg per liter 2,4-D. And for multiple shoot induction from nodal explants, it is 2 mg per liter BAP and 0.5 mg per liter NAA. Future studies includes enhancing the production of secondary metabolites through hairy root cultures. In the present study, young nodal explants from 1 month old grown hemidesmus indicus were used in vitro regeneration. 3 types of growth regulators in different combinations were tested for multiple shoot formation and callus induction. A combination of MS media along with 1 mg BAP and 0.1 mg 2,4-D. When 2,4-D is added to the leaf disc, the highest proportion of callus induction was observed. Proliferation of shoot was seen with a combination of MS along with 2 mg per liter BAP and 0.5 mg NAA. Once the regeneration protocol is optimized, hairy root culture induction will be carried by infecting the Agrobacterium rhizogens. So I need to continue my work, and these are the references. Thank you for giving me this opportunity. Hello. Hello.
Yeah. Hello.
Hello.
Ma'am, can you stop the PPT sharing, ma'am?
Okay.
Okay, thank you, thank you.
Okay.
A very pleasant good evening to distinguished guests online and offline, and my dear fellow students. I'm Divya Double Party from 3rd BSc in DYC. So right now I'm doing my research project, my hobbies research. On this project, I would like to introduce in my biology of Raju sir. So I'll try to keep this short and sweet. the main theme of our conference is to attain sustainable future through transformative technology. So, production of drug has been rapid these days, day by day, and as much as we produce these drugs, it is also important to dispose them properly. So my work is going to be about how to dispose it properly to achieve a sustainable future and growth. So this is the basic point of my work. First one is, if we do not dispose our drugs properly, it poses a serious threat to our environment. Not only developing resistance of microorganisms, but it will also affect our environment, that is through water pollution, soil pollution, etc. Morning we have already seen how serious antifungal disease can be and how life-threatening it can be. So it is important to develop our drugs, and also it is important to dispose it properly so that these microorganisms will not develop the resistance against our drugs. And also the research going on these fields, particularly antifungal, is comparatively low because because of lack of awareness, and also these cells are similar to our eukaryotic cells. That is why it is hard to find a suitable drug. So, with this study, we'll also not only check the degradation and also we'll do the analysis by using HPLC and other analytical methods. First one is we'll try to identify chemical methods. We have tried to use different chemicals and mix it together with different concentrations to see how that particular chemical combination is needed in this particular drug. And then we have analyzed how much of the drug is degraded, and then we did the antimicrobial test. That is our objectives. The methodology we have followed is first one we have this mental reagent. It is FDCl and H2O2 combination. These are comparatively of cheaper cost, so we can use them to in large scale to degrade drugs. And the next one is autoclaving. We are familiar with it in many of our science experiments at 15 lbs pressure and 121 degrees Celsius. For 15 to 20 minutes, we use it more to degrade or to achieve sterilization. Here we have also used this method. We also SGS treatment, which is anionic surfactant. It reduces the surface tension and also increases the intermolecular bonding between the drug molecules, and also UV radiation. And the HPLC analysis. About HPLC analysis and microbial activity, I'll be speaking about in the coming slides. HPLC analysis. So this is the result of our work. These peaks is shown. We have multiple peaks. That means our drug is degraded into simple forms. From the complex form, it is degraded into simple forms. So we will observe multiple peaks. Based on this peak, we will determine if our drug is degraded properly or not. So our drug is almost degraded pretty well. So this is microbial activity. Few of us are familiar with this. So with this method, we have tried to understand if our drug is properly disposed. So this is the culture. So if our drug is completely degraded, we cannot find anything around here, which means that our drug cannot work on this particular microorganism. And if the drug is not degraded properly, we can observe this zone of inhibition, which means our drug is not degraded and the microorganisms are affected with this drug. So here I'm doing, this is my photo of me doing that antimicrobial activity. Key findings, mental reagent, we use this method. It degraded pretty well the components. And also antimicrobial activity is also shown in comparatively reduced form. And also it can be used for large scale quantities. We've also heard that our lab work or our knowledge need to be transformative. So we are hoping that this method can be used in large scale towards taking steps for sustainable future. Conclusion is that this mental reagent method we worked on is working well to neutralize the antifungal drugs, and it also neutralize microbial activity because, as Monica already heard, it is really important to dispose the drugs properly. If not, these antifungal drugs are present, abundantly in our environment. So if we not dispose it properly, these develop resistance against these particular drugs, and these drugs cannot be used to control that particular disease caused by these microorganisms. Recommendations, what I humbly would like to recommend is that we can implement these fentanyl reagent for disposing in large quantities. A lot of drugs are produced and also a lot of drugs are not being used or they are not being marketed. We can dispose them by using this method in large scale. And also we can extend this drug not only for antifungal research but also we can use this method for other drugs also. Inside this we cannot say it today, but we can call it experiment. We can try to mix different chemicals for a better output with some guidance from our faculty. And also we are students, right? So we may not have that equipment, so we can collaborate with pharmaceuticals or NGOs who are trying for this better future or this research work. We can collaborate with them to do a better work. Then I thank Almighty God firstly for His grace, and then my heartfelt thanks goes to my guide Dr. Ramraju sir for his support and guidance throughout, and the Department of Biotechnology and Documents. Thank you. These are the quotations I've searched to encourage you guys to ask questions. That is, questions are more important than answers, and question everything, including your assumptions. With questioning, you can develop your answering and maybe find a better future.
Thank you so much, Ms. Vijayashree, Ms. Secretary. Now the floor is open for your question and answer. Dear participants, if you have any questions, you can okay. Everything is clear for the students. Now we can thank Vijayashree and call next participant.
Thank you, Vijayashree. Now I would like to invite our next presenter, Ms. Blessy, to present her paper entitled Screening of Locally Adapted Chili Genotypes for Drought Tolerance at Seedling Stage.
Good morning, everyone. I'm Blessy, pursuing molecular biology MSc in Central University of Andhra Pradesh. I'm here to present my research work, which was screening of locally adapted chili genotypes for drought tolerance at seedling stage. We have done this project under the guidance of Dr. Jaina Kumar Naik sir, and he is our assistant professor. And we have selected this genotype. It is capsicum annulum, and we selected eight genotypes of chili, and we made our research studies on that eight genotypes, and we screened one of the genotypes which was drought tolerant among eight. Is a member of Solanaceae family which was highly sensitive to drought. And first of all, I want to tell that what is meant by drought stress. And stress is mainly two types: biotic and abiotic stress. Biotic is nothing but which is internally which was occurred. It was called as biotic stress, and abiotic stress is nothing but which was environmental factors are affecting the plant to grow and its reproducing and growth and development. It causes the abiotic stress, and the drought is one of the abiotic stress. The impact of drought is nothing, maybe soil water availability was very less, and this makes the plant to grow. Effect of plant growth is very less, and plant growth may be affected at two stages. One is germination stage and the seedling development stage.
And we selected our 8 genotypes at the seedling development stage. And also, the impact of drought may be at changes in morphology and physiology of the plant and metabolism of plant and also physiology of the plant. And not only the plant metabolism and physiology and biochemical functions, it also affects stomatal functions of the plant. Our research aims to selection of tolerant genotypes and improve understanding of oxidative stress under drought conditions. Whenever the plant is lack of water, it may generate the ROS, reactive oxygen species, which may affect the plant entire metabolism, and it causes the production and yield may be very low. Next slide. And these are the 8 genotypes we have selected for our research work of chili. Among 3, we have sold in our fields, and we among 8 genotypes we selectively chosen and we irrigated first of all for 45 days of irrigation, and we screened 3 genotypes after 45 days and for 6 days. After 45 days of irrigation, after 6 days, we maintained the stress. We imposed the stress to the 8 genotypes, and we screened 3 genotypes from 6 days of stress. And the 3 genotypes are tolerant to the 6 days stress. The 3 genotypes are Mongolian 5575 and Lucky 2 and Tamana. These 3 are genotypes which were tolerant to 6 days stress. Next. And random block design was for our experiment we have chosen the random block design, and we prepared our field under the red soil from December to February. And we screened 3 varieties of chili, and we made again 6 days stress to the 3 genotypes. And among the three genotypes, we tested some of the parameters like chlorophyll content and also electrolyte leakage. These are the parameters we took for the screening of that 3 genotypes after the 12 days stress. I mean, first 6 days stress and also adding the 3 genotypes and again the 6 days stress. Total, it is 12 days stress. After the sowing of plant, after 45 days of plant irrigation and 6 days of first stress and 6 days of second stress. Totally 57th day, we performed some of the parameters to check shoot length and morphological and physical characters like shoot length and root length and electrolyte leakage, chlorophyll content and proline content. And we made one of the best drought tolerant chili genotype. Next slide, sir. Root to shoot weight ratio was calculated by using dry weight, and the relative water content. We calculated the relative water content by using the formula fresh leaf weight minus dry leaf weight by turgid leaf weight minus dry leaf weight. Fresh leaf weight was calculated after the 57 days of sowing, and after sowing, 57th day we extracted samples and we made to the leaf to turgid by keeping it in distilled water, and we calculated the relative water content. And SPAD chlorophyll meter reading. It is the test we conducted to measure the chlorophyll content before and after the stress tolerant genotypes we performed, and controlled plants and stress imposed plants. 2 types of plants we have for our reference. Next slide. Electrolyte leakage. This is the another parameter we checked apart from shoot to root wavelength. And cell damage was calculated by using the electrolyte leakage. When a plant is imposed to stress, it may the cell walls of the plant material may weaken. It may lead to electrolyte expulsion
apart from cell walls. Weakening of cell walls may take place due to water availability will be less. We calculated electrolyte leakage by using the formula electrolyte leakage is equal to reading 1 by reading 2. And these readings are taken from dried plant, and also the after distilled water we keep another sample, and that is the turgid one. And both readings are took, and we perform electrolyte leakage parameter. Specific leak area. By using one of the leaf area meter, we specific leaf area was calculated. And the chlorine content was the last parameter which was conducted calorimetrically by using absorption of chlorine. By comparing standard deviation, standard curve, and we made the proline content, how much proline content is present in controlled plants and also stress imposed plants. Next slide. After analyzing the all parameters, we prepared a statistical analysis data, and we made some of tests by using ANOVA test statistic test software. This was a software we used to check all the parameters which was created by manually by ourselves. And these are our results which was analyzed by statistics. And total plant height, shoot length, root length, and shoot dry weight. Among all the three genotypes, the Napoleon 5575 exhibited the high drought tolerance when compared to Lucky 2 and Tamana. And relative water content and electrolyte leakage. Total chlorophyll and total product. Here, in electrolyte leakage, the Napoleon 5575 exhibited high electrolyte leakage when compared to other two genotypes. Here, the electrolyte leakage will be exceeding when compared to controlled plants, because in controlled plants, obviously the water availability will be very high because we irrigate every day water to the plant. When coming to stress imposed plants, we will avoid irrigation of water to the plant. Then the cell walls and the cell membranes will weaken, so that electrolytes which are present in cell will be expelled outside. So that electrolyte leakage will be more in stress imposed plants. That was the graph showing that only. Here, the electrolyte leakage of stress imposed plants are high compared to controlled plants. Remaining all parameters will be taken as which will be lesser than in every parameter. The controlled plant is having higher parameter range when compared to our stress imposed plants. By analyzing all the statistical By using all the statistical tools, we made conclusions that Napoleon 5575 was the one of the best drought tolerant plant, which was helps the farmer to cultivate in the crops, and to they increase the production of yield. We made a survey in our Anantapur district, which was a very water content will be very less, and there may be high prone to drought containing areas. Here the chili genotype, that was Napoleon 5575, was the best what we can say best genotype. We can use that genotype in our fields, and we can improve our crop yield and production. And this suggestion was given to Anantapur district farmers, and this was the initial step took by our Central University of Andhra Pradesh Molecular Biology Department students, and higher next upcoming projects are done under the guidance of Dr. Jayana Nayak sir on the chili genotypes on molecular level. Thank you for giving me this golden opportunity. If you have any queries, you can ask me. Any questions now?
Thank you, Ms. Lucy. It is now my pleasure to introduce a special session on career guidance for studying abroad, sponsored by I-20 Fever, the proud sponsor of today's conference. I-20 Fever is dedicated to helping students navigate the journey of pursuing education abroad, offering invaluable advice and support for those aspiring to study in international institutions. I invite the representative from I-20 Fever to take the stage and begin the session. We encourage all the participants to actively engage themselves in the session. Thank you. I invite Vaishnavi to welcome Sir with a sapling.
Stella.
Thank you, madam. Thank you, sir.
Hello everyone. Good afternoon, first of all. And myself, let me introduce to my colleague, that is Yami. I'm working as an USA and Australia counselor from I-20 Fever. Um, just in this presentation, we will just give you brief introduction about the countries which every student want to do masters or higher education in abroad. So we will just give you brief introduction about the countries which you want to do. So let's start. And before starting, just we will introduce ourselves. Actually, I have introduced you, that is Yami, and working as a counselor. But I want to just introduce our organization, what services we are going to do, and what services we have done up. So it's a basic introduction about study abroad and the country introduction, and then about the student profile and some question and answer session. So about I-20 Fever. It's our brand name, I-20 Fever, and our registration name was Yatapo Consulting Private Limited. And we are since 2006, and we are up to now. Uh, just we are still continuing 2025, and we also happily send 33 plus students, 33,000 students, sorry for the term. And we are send these 33 plus thousand students to different destinations: USA, UK, Ireland, Australia, France, and Germany. So this is our journey. And what we offer. So not only a service oriented, we also offer a coaching, which was different examinations you have to take before applying for a masters in abroad. So for some countries you need GRE, for other countries maximum you need an English proficiency score, that is Duolingo, TOEFL, IELTS, and PTA for Australia, and SAT for the UG. So we offer these at a lower price than other consultancy. We offer them online itself, and we also provide books also for the reference. And And even if you're keen about this particular coaching, and we'll also provide it as a free also. Okay. So next, so let's begin about introduction of the countries. Okay. Yeah. In this study abroad program presentation, I will explain you about these following countries: USA, Canada, Ireland, UK, Australia, France, and Germany. We deal with these uh, countries as well. So, yes. Coming about a brief about USA, everyone know about USA. So USA is an what we'll say land of opportunities. Event failure USA అందరికీ తెలుసా yes so how many of your cousins relatives are over USA I think everyone most probably all of you just give me raise your hand it's enough so plenty of you you're all, oh, okay almost all of the each and every student had someone related to US right so it's a well-known destination for everyone for your abroad studies. So what are the USPs? What opportunities you will get while studying in USA? I want to just give a brief about that. Okay. So US is a top is a country with the top schools, top universities, and also it is a land of opportunities a course you take you pick a course you will have a institution in US where it provides it and also you will have similar industries related to that course also so that without any industry you can't sustain after pursuing your masters right so you just had each and every industry for you to sustain after your masters so next what are the top reasons to study in USA? The first thing is its visa period. Okay. For it has 5 years of stamping. You will get 5 years of stamping, 2 years for your masters, and rest 3 years. That is 36 months for your extension which you can work full time. If you get a full time, you can directly apply H1B. If you get picked, you can work. Once if you get H1B, it's very easy to get settled over there because after that extension would be very clean and easy also. And trust me, US is a country which pay high for software or biotechnology, whatever stream. US will be high in paying. Okay and then so these are the popular courses for your biotechnology students. You can have a bioinformatics, biotechnology, epidemiology, or the biomedical science, each and every. And you also have related industries which offer job opportunities after pursuing masters in this particular degrees. So US is a full factored land. Okay, and then so I'm just wrapping up USA. I'm going to the other country that is Australia. Yeah. So Australia is the second destination of every student who want to do their masters or abroad because Australia is the place where it is a double of India, and with low population, lesser population. So Australia does not easily allow each and every student to study over there. They genuinely see quality over than quantity. So Australia is a country where you can have a health sector is very good in Australia. Health and mining both are good in Australia. So and that too and in Australia you are legal to work 20 hours a week. Legally work part time 20 hours a week and during your holidays you can work 40 hours a week. So it is legal to work part time where in other countries some of the countries like US it is illegal. So you can have a work opportunity also. And then the main thing about Australia is we no need to apply for scholarship separately. The application you apply for the admission also, they will calculate scholarship and then they will give you a scholarship at the time of admission itself. It's a plus point with Australia. Yeah. And moreover, Australia has G8 group of 8 universities which are equivalent to Stanford, Harvard in USA. So these group of 8 universities would be a cost of 60 lakhs, 40 lakhs, 50 lakhs, but they are as equivalent to Stanford University, which are year 1 CR tuition fee. So and also yeah, getting to the extension visa, it gets 2 years for your masters, and then if you study in regional area, you will get 3 to 4 years of extension. That is total 7 years of visa. So if you study in regional area, you will have a points for your PR also. If you read in a regional area, then you will have extra points for your PR. If you stay in nearly 6 to 7 years and you have full time employment and a sponsor for you, then you will eligible for applying PR, and that is too quick and easy to get a PR. Whereas in US, it needs 15 years, 20 years to get a green card. And also, the popular courses in Australia, first thing is agriculture and marine, and then biotechnology, and other food, nutrition, and biomedical sciences. And trust me, health sector is too good in Australia because Australia prefers health as first, demanding the next. Okay? Um, yes. And my fellow mate, Swarup, will continue from here for further countries. Thank you.
Good afternoon, everyone. I'm Swarup, and I'm the UK and Ireland Council of So I'm in many countries. First, we have Canada. So, how many of you know Canada? Just quick. Thank you. So, it's a neighboring country of US, right? So, it's more or less the same as US. You get to chance to study for 2 years of master's degree, and after that, you will get PSW for 3 years, in which if you are able to crack a green level job for 1 year, that's it. You are eligible for PR. That's the most biggest advantage for Canada. And second, and most of the North Indians prefer Canada for easy settlement because it's one of the easiest countries to get PR and one of the major sectors in which Canada is popular is like food because in Canada the weather is very cold and they do not depend upon agriculture. They prefer meat over rice or other foods. So canned foods are preferred most in Canada. So for students like food technology or like you biotechnology students, it is very good. And you can see the popular courses MSc in agriculture, MSc in food technology, MSc in biotechnology, food science and technology, etc. As I said before, for food processing and other, these are the major industries and the companies which offer for major jobs for students in Canada. Coming to the UK, it is one of the most dream destination for students and one of the easiest countries to land and pursue a master's degree. So one good thing about UK is you finish your master's degree within 1 year, and you get transfer. You get PSW for 2 years, and within these 2 years, if you are able to crack a tier 2 job and continue the same for next 5 years, then you are eligible for the PR in UK. And another good point about UK is you can work legally part time during your class, and you can work full time, that is 40 hours a week during the summer break or semester breaks. Even during the breaks, you can earn up to 2 lakhs a month. Coming to the course, UK has one of the top universities. I think most of you might have heard about the Oxford University, correct? Where is it? It's in the UK, right? So, so one of the top, I mean, the leading universities are in the UK, and even UK universities like Cherry University, these are from 19th century. They are offering different kind of courses since 19th century and it still continues. So UK's education system is most dependent on the healthcare sector. Whoever wants to pursue their master's degree in health, they can easily opt for a master's degree in UK. Yes, as I told before, 1 year, so within 1 year you will be able to finish your masters, and it is as low as 15 lakhs. In some cases, it's even 12 lakhs. Within 12 lakhs, you can finish your master's degree in UK. One more advantage for UK is the universities revise the syllabus every year. Not only one university, every university in the UK they revise syllabus according to the latest technology and latest developments. And these are the popular courses, as you can see. So, like, there are different types of courses like research and development, clinical research associates, different aspects and so on. And you can see these are the industries that offer jobs for international students. This one. And coming to Ireland, how many of you know Ireland? Have you ever heard about the country Ireland? Where is it? Okay, which part of the world is it? Sorry? Correct. It's part of European nation. It's just adjacent to the UK, and it is similar to the UK. You can finish your masters in 1 year, and you can even finish your masters in 10 lakhs starting, and you'll get chance the same as UK. 2 years of PSW, you get 2 years of PSW, and if you get a job in 2 years and get it extended for another 2 years, within 5 years you'll be able to get a PR. And Ireland is one of the safest countries. You can see it's the third safest country in the world, and in the past 25 years there is no record of crime. It's the safest country for women. And it is one of the fastest growing economy in the European nation. And if you have a student, how many of you like traveling? Good. Then it's a proper destination for you all. If you have a Schengen visa, you can travel around 28 countries with this visa. I think that will be a good asset for you. Right? Next one. And one more USP for Ireland is it is the one and only English-speaking European nation. The rest all countries they prefer mostly European languages like German and all. This is the only country that allows English. If you are able to crack the PR and after that if you get a citizenship, it's the fourth most powerful passport. And one more advantage for Ireland is the lowest tax. Have you, do you have any idea how much tax we are paying in India? What is the range? If your income is about 25 lakhs or 30 lakhs, we pay around 28% of our income, right? But in Ireland, the tax is up to 9%. Maximum would be 18%. So that is one big advantage. And you name any of the top companies like Google or Pfizer is one of the medical company. All these companies they have their second headquarters in Europe. That is, specifically in Ireland because of the tax exemption. And it is one of the top destination for pharma and medical students. I mean, even biotechnology students, you can prefer this for your masters, as it is one of the best country in the world to finish your. You can see these are the popular courses: biomedical sciences, biotechnology, food and science. And one more advantage is these courses keep changing every year, and they revise every year. They are now even adding technology to these courses like food technology and other. So, That's brief about all the countries. Now you might be having a doubt like how should we start? What is the procedure? And what are the documents we all require? Correct? You must be having these docs. So it's simple as that. So all you need is if you finish your bachelor's, just you come to us with all your documents like all your mark sheets, provisional certificates, ODs, and all. That's all you need. I think you don't have any backlog come. Do you? Good thing. Yes. Previous ones. So one more advantage is like you need an English test to perform well. So write at least any one of these English tests like Duolingo, IELTS, TOEFL, or PTE, so that it will be advantage for you. And coming to the other documents, you need letter of recommendations from your college and statement of purpose, which states your intention to study masters in abroad, and of course bank documents. So you must be worrying about the financials and all. You need not worry. We have direct tie-up with HDFC Credila, and a representative from the bank always stays in our office to assist you with your financials. If you are eligible for the loan, they will provide education loan up to 40 lakhs. Excellent. So this is in a nutshell. This is the process for all the countries. You may take a snap if you want. Will be there for 1 minute. So overall, it's simple. You apply to the universities, you get the admission, you pay the tuition fee, you apply for the visa, and that's it. You can fly. Can we proceed further? Okay. Good that you are showing interest. Someone from the behind are capturing. Shall we? Okay. Next one, please. Okay. First, you might be thinking, we are from normal background, can we go abroad? Stop thinking and stop putting it to yourself. Believe in yourself, make a step, move further, and we are here to assist you. So that's on our side. Do you have any questions from your end? I need at least 3 questions, please. Let me know whether you have listened to us or any 3 questions. No? All good? So you must be knowing about every country, right? Fine. Next one, please. So we are from I-20 fever. So she has a question. Okay, please. Sorry? Forensic sciences. For every, like UK and Ireland is best for forensic sciences. It's one of the best. I mean, these two countries are budget-friendly countries. You can finish your masters in 10 to 15 lakhs. Canada, it will take 2 years to finish your masters. It is also good, but I suggest you to go for UK or Ireland. Any other questions? Please take the mic from me.
So if we want to, can we like extend our masters for 2 years in Ireland and UK? Or is it a compulsion that it's only 1 year?
No, for Ireland it's 1 year. Mostly the process of 1 year. You have option to do masters for 2 years in the UK. First year it will be the theoretical classes. Second year it is like internship. You go for it. For UK you have the option for 2 years to do your masters. In the same period of time for PSW. For these either countries, you will study master for 1 year and you will get 2 years of PSW. Thank you. One more. someone from previous presentation, they said you will move ahead only if you ask questions. Okay, then thank you. Shall we move ahead? Okay, this is our address, I-22 Vijayawada, Daily Manor Lane, opposite Amity Silks. Yes, if you want, you can take a screenshot or else. And these are the details of our branch head, Mr. Kishore Babu Kancharla. That is the phone number. Please make a note of it. And I think you must have taken the kids from us since we are here yesterday and today also. How many of you have taken kids from us? Great. If you want, you can. We'll be there for here for another 1 hour. Please come to us and take the kid. Next one, please. Yes. As I suggested, for you biotechnology students, UK and Ireland are the best destinations. So that we are conducting an education fair for UK, Ireland, and Germany on March 23rd. That is on Sunday at our office itself. You can see the address. And these are the universities that are going to visit. Direct representatives from universities will visit our office on that day, and even white faces will be coming so that they will directly answer to you. If you have any doubt, that's my mobile number. You can contact me anytime. I'll guide you. That's it. Thank you for giving us this opportunity. And I extend my thanks to Dr. Rekha ma'am and Dr. Gangaraju sir. Thank you so much, ma'am. Thank you, sir. Thank you, and have a nice day.
Thank you, sir, for the informative session. So now we will proceed for tea break. Kindly please come by 4 p.m. sharply. Thank you.
Truth and charity, and shine as beacons of hope and change in the world.
Education is the most powerful weapon which you can use to change the world.
I extend my heartfelt congratulations to the entire Ministerial family for achieving yet another remarkable milestone in your journey of excellency, empowerment, and enlightenment.
And the corporate experience and trained corporate institute organizations, Antari Auditor, and competent teams, and finally a very
ఆ మేడంతో చెప్పండి, బయటకు తీసి రమ్మని చెప్పండి. బయట బయట
Which you can use
Excellent. Heartfelt congratulations to the principal of achieving yet another remarkable milestone in your journey of excellence, involvement, and enlightenment. congratulations to the entire Principal of Sandy for achieving yet another remarkable milestone in your journey of excellence, involvement, and enlightenment. My excellent, my heartfelt congratulations on achieving
Education is the most powerful weapon which we can use to change the world.
Esteemed, my heartfelt congratulations to the entire vegetable family achieving yet another remarkable milestone in your of excellence, empowerment, and enlightenment.
Education is the most powerful weapon which we can use to change the world.
I extend my heartfelt congratulations to the entire vegetable family achieving yet another remarkable milestone in your of excellence, empowerment, and enlightenment.
Good evening, everyone. We now move forward to oral session 4. We are delighted to welcome all the participants who will be presenting their research and review papers during this session. Your contributions are imperative and essential. I now request Jasmine Kesana Kurti to present her paper entitled Robotic Harvester.
Good evening, everyone. Good evening, everyone. This is Jasmine Kesikuti from Agriculture and Rural Development. I'm presently in second year. Today, I want to present before you about the robotic harvester, which is an essential component for the whole agriculture sector. As we see here, sorry, we see here challenges in traditional harvesting methods. Farmers face many problems in while doing the traditional farming, whereas they face the low productivity in the yield and the consuming many health issues, injuries while working in the farms as well. As we see, traditional methods can be limited by weather conditions, terrain, and crop complexity, make it difficult to harvest the crops. As well, it impact the profitability and economic value and decreases the many potential levels as too. Next slide. As we comes here, robotic harvester, which is a automation of artificial intelligence, whereas it derives from the many features of computer vision, AI-driven algorithms as true. Whereas within the ability 24 by 7, it works continuously in any places of farm regions in agriculture sectors. It works in wetlands, drylands, and greenhouses, and many places as well of with the warehouses and too. And its productivity in fruits, vegetables, and many cereal crops, grains, and etc. Next. As here comes to the key components of the robotic harvester, there are sensor cameras which detect and locate the specific fruit area and take the pictures, and it shows the whether it is a defect with any disease or not. And it comes to computer vision where it shows color and fruit size and the perfection of the fruit shape and those. And here with the robotic arm, it is the human hand where they mechanically grip detaches the fruit and without causing any damages. And here is the navigation system where
use of GPSC transactions where robotic insert with the GPSC track where we'll use in the car GPS and any locations as well. Next. As we come here, benefits of robotic harvester where increase efficiency, where it works faster than the humans. And improved accuracy, it picks up the fruit with best quality without damaging the fruits. And the enhanced safety, it has easy handling and without weightage. And the increased flexibility, it works by 24/7 according to the field conditions. And reduce labor costs, their money and efforts also. And improve crop monitoring, it detects the disease efficiently. And environment benefits, it conserve water and reduce the wastage. And increase scalability, which can apply many resources to detect the disease and solve and give information to the farmers. And improve data analysis and gives data perfectly whether it is working or not and with many specifications. And enhance food safety, it gives the better quality foods without contaminations. And applications of robotic harvester which reduce the labor costs and enhance crop quality, minimizes the losses and automates tasks and livestock and reduces the waste and supports various types of harvesting in fruits, vegetables, cereals, grains, and many etc. Next. Where it comes to the challenges in the robotic harvester, as we see, robotic harvesters which are derived from the artificial intelligence, they are cost effective, but it is efficient to use in many areas. It increases the productivity, yield attributes, and many others. As it needs the skilled technicians to operate perfectly, and the many training programs should be ensured to the farmers as well. Next, please. As here is the future developing and trends in the robotic harvesters, as in advancement in AI and machines which creates the best productivity. AI machines and learning integrations with other agricultural productivity, where it creates a increased focus and sustainability with the environmental impact, advancement in sensor technologies and data analysis, and increased adoptions in emerging markets and business strategies. As we come to the conclusion, robotic harvesters are revolutionizing and ensuring global food security and sustainability. By embracing this innovative technology, we can create a global food security and create a better, efficient, productive, sustainable food system for the future generations. Thank you.
Thank you, Ms. Jasmine. Now I invite the next presenter, Ms. P. Srihita, to present her paper entitled Crop Disease Detection Using Artificial Intelligence.
Let me introduce myself to you. My name is Stella. I am from Agriculture BSc second year. Today my topic is based on crop disease detection through AI. Nowadays, farmers facing mostly the problems based on the diseases. Due to diseases, the farmers face crop losses. According to the FAO, there is financial losses of
Srihita. Mail section. Mail. గంగరాజ్ సార్ ఏమని పెట్టారు అది శ్రీత్ అక్కడ చూసారు సో వన్ సెర్చ్ సార్ ఇఫ్ దట్ నేమ్ ఇస్ లైక్ వెరీ శ్రీహిత స్టిల్ ఇస్ ఆర్ RIHITH I think it's not there.
Next person,
Next to some sir that names. Kanti Punita, I think.
This one. Kanti Punita.
I think this one, sir. Wait.
You can proceed to the next person. You will send to me. Punita అయితే కాంతి పులిత
కాదు సార్ శ్రీత కాదు సార్ మెయిల్ రాలేదు సార్ అసలు
Miss K. Kandipulita to present her paper entitled Organ-on-Chip Technology in Insulin Production.
Good evening, everyone. So, my review paper which I have written was on organ-on-chip technology in insulin production. So basically, organ-on-chip technology is an in vitro method of culturing your cells. So it can also be used in personalized medicine. Like we collect the cells from the patient and we culture them outside. And basically, how it is related to insulin is we take the inactive or deactivated pancreas beta cells and we culture them outside so that we could again produce insulin. So after culturing these cells, using microfluidic systems, we're gonna insert them back into the host so that his pancreas could work properly and produce proper insulin. So I've researched a few papers, and that I've seen like other organ on chips, like liver on chip, lung on chip, heart on chip, kidney on chip, and intestine on chip, and also multi-organ on chip, and it is also called a human on chip. So I thought it would be better. Why not use it for diabetes? Because in the recent times, a lot of people are facing trouble with diabetes. So this is my presentation. I thought this would be better. So this organ-on-chip technology, and this we use microfluidic systems. These microfluidic systems, in these we insert the cultured cells. So this is all integrated into a small microchip, and this microchip could be implanted into the host. And I would like to say a few points about the other organ-on-chips. Liver on chip. It is the first organ on chip discovered, and when it comes to lung on chip, it works on gas exchange medium, and heart on chip it is like made from a PDMS, a stretchy material, and kidney on chip because kidney is the most important organ in the body, which helps us to maintain our body balance.
It is performed on mice kidney cells, and the intestine on chip. It is done by culturing the CaCO2 cells from our intestines, and the multi-organ on chip, as I've told, it mainly concentrates on bile acid management. So this is my presentation. Thank you.
It's working now. But range, that's the same.
Thank you, Ms. Kandipunita. Now I request Ms. P. Srihita to present her paper entitled Crop Disease Detecting Using AI.
Let me introduce myself to you. My name is Srita Tatsetti. I'm from Agriculture BSc second year. Today my topic is based on crop disease reduction through AI. Farmers facing nowadays lot of problems due to crop loss and crop diseases. The FAO estimated that there is a total loss of crop diseases. How AI transforming is agriculture, learn about this role in crop disease reduction, and this presentation will explore the benefits, the future of impact on global food security.
Share this one.
Next slide. The global food security system faces many financial losses nowadays due to the crop losses, and the farmers also faces lot of challenges because of crop diseases. Hence, this AI helps to detect the crop disease and enhances the precision agriculture. The problem is The problem is early disease detection is essential for the maintaining crop yields and ensuring global food security. Traditional methods are often time-consuming and prone to error. Hence, this AI artificial intelligence offers a powerful solution enabling rapid, accurate, and scalable diseases. The presentation overview will explore the current state of crop disease detection and in special AI techniques. Crop diseases causes sustainable losses every year. There will be $220 billion annually. And majorly significant crops like rice, maize, wheat are reducing yield 17 to 30%. And due to some climatic changes and weather variables also, there is the loss of crop yield. The high temperatures and heavy rainfall conditions causes the crop loss. And examples of the devastating crop diseases include the wheat rust, rice blast, and late blight in potatoes. These are the entire crops have far-reaching economical and social consequences. Next slide. Machine learning and the deep learning are the two methods of AI which help us to understand the disease detection. It is very rare. It is helps to farmers to understand in the easy way. And computer vision AI algorithms helps to identify the disease the images the AI algorithms capture the images symptoms of diseases including discoloration characteristics lesions and signs. Training data AI models assist the data is fixed in the AI models to help the farmers. Early and precision detection. AI crop systems can detect diseases at early stages before the farmers can be seen before the naked eye. Next slide. Data is the key. It is one of the important steps in the AI models. Data can process the images through image sources. The image sources are in the red, blue, green, and hyperspectral and thermal. And each type of image really provides an image of that disease. And public data sets. Public data sets helps the farmers basically according to the environment. This public data sets changes according to the surrounding environment. so that they can relay the disease of the public data and collection methods. The collection methods are one of the important step in the data, and these collection methods images can be acquired using drones, satellites, and smartphones and field sensors. And data augmentation techniques such as data augmentation are used to increase the size and diversify of the crops. Next slide. These are the 4 steps of AI models: conventional neural network, transfer learning, object detection, and segmentation of the 4 steps of AI models. Next slide. Recently in 2022, the Australia has faced a lot of the Australia of the farmers faced the crop disease in wheat rust, and they Western Australia during 2022 this problem has faced it. The project utilizes satellite imagery monitor to large wheat fields, and the machine learning models help the farmers to detect the disease and capture at the moment. Next. Real world applications on the platforms. The farmers, all of the farmers nowadays having smartphones, and in the smartphones are having agro doctor apps. And through that agro doctor apps, the farmers can detect the disease easily and eradicate in early time. And next one, drone-based services are also provided by the government. Through these drones, easily accessible of nowadays, the government also providing funds for the drones to the women basically, and these drones capture the diseases of crops and help the farmers in eradication. And cloud-based platforms such as Microsoft FarmBeats and IBM also help to detect the diseases. Challenges and the future directions. Data scarcity. Rarely diseases. The farmers cannot overcome data scarcity, especially in diseases. Obtaining sufficient high-quality images and training AI models can be difficult. The module robustness also not yet developed till in the AI. Explainable AI. Explainable AI. The farmer should keep trust on the AI, hence it is helpful to them. And integration with technologies. Integrating AI with other technology like log services, sensors, precision spraying can help the farmers. And my conclusion: AI-powered crop diseases detection helps for the transformative technology for sustainable agriculture and helps the farmers the minimal use of fertilizers and pesticides. It helps the early detection of crops and diseases.
Thank you, Ms. Srilika. Now I request Sheikh Rakia Sultana to present her paper entitled Self-Regulating Smart Irrigation System.
Good evening to you all. This is Sheikh Rakia Sultana from third year Agriculture and Rural Development. My topic is self-regulating smart irrigation system. This is a technology-driven agriculture irrigation. It flows the water automatically. This works with the sensors and monitors soil moisture, and the weather temperature can be automatically operated by using these soil moistures. The objectives are mainly water conservation, automatically piping, and it also observes the crop health, and also energy efficiency and climate adaptability. The components that we have used in this system are battery, Arduino relay, and soil sensor. How this system actually works is the sensor absorbs the soil moisture level, automatically it operates, and it sends the data to the Arduino. Then the Arduino sends the signals to the relay module. After sending the signals, the water is supplied automatically. When the soil is in dry condition, the water automatically stops. When it is in wet condition, it gives the water. This is the model we have presented in Horti Expo 2025 from the Agriculture Department. This is the soil sensor and the water pumps. When the soil condition is in dry condition, the water automatically releases. When it is wet, it stops. Power supply. Power supply is supplied by the batteries. There are two types of batteries. For small field, actually we will use 9 volts of battery, and for the large fields we use 12 volts. In the many agriculture fields, normally farmers are not able to apply more current supply to the fields, so that they can also use the solar sensors. This is the moisture sensor. After giving the connection, the volume and the moisture will applied on the screen. This is the connection. The motor is connected to the tank, and from that it is connected to the Arduino to the relay mode. For future enhancement, this model we have displayed in our agriculture 2025 in our college. But for the future, we are thinking to develop the model by applying the multiple sensors, and also by using the mobile apps, and also by forecasting apps. And also this system is useful for the home gardens, agriculture fields, as well as smart city landscaping. And this system mostly useful for the farmers because in many villages they are lacking of current supply, so that without the current supply they can use the sensors also for the power. Thank you.
Thank you, Ms. Satya. Now I invite M. Chandrika to present her paper entitled Role of Tissue Culture in Genomic Conservation. I hope everyone would like to run to your destinations, but kindly bear me for about 10 minutes. Sunset greetings to everyone. This is Chandrika Malla from 4th BS Agriculture and Rural Development and from Stella College. First of all, I would like to thank the Department of Biotechnology for giving me this opportunity. I would like to present my article on role of tissue culture in genomic conservation. Slide, please. Coming to the detailed topic, detailed presentation. The conservation genetic diversity has increasingly recognized as a keystone towards the production of biodiversity and natural resources for future generations of humankind. Biodiversity faces numerous threats, including habitat destruction, climatic change, pollution, leading to rapid loss of species in the world. In response to these challenges, plant tissue culture has emerged as a powerful tool in ex situ conservation, offering unique advancements in preservation and propagation of rare and endangered plant species. Let us explore more about role of tissue culture. Next slide, please. It is the most frequent technique used in biotechnology for basic and applied purposes, from investigation of plant development processes, functional gene studies, micropropagation, and crop improvement to rescue threatened and endangered plant species with high quality, healthy plant materials. The PDC was initially developed to demonstrate the totipotency of plant cells, which was predicted by Gottlieb Harbeland in 1902. He was an Australian botanist. Thank you. So, welcome to the. There are many approaches which could be done in plant tissue culture.
where I have mostly focused on cryopreservation technique. It is an alternative storage method for exceptional species like oats, orchids, mulberry that produce few seeds or the seeds which are intolerant to drying or freezing. Cryopreservation is a process by cooling and storing the plants or else tissues or organs in liquid nitrogen for about minus 196 degrees Celsius. Currently, it is the most innovative and affordable biotechnological approach that allows long-term conservation without the risk of genetic modification. Next slide, please. Next slide. Thank you. At this point, I would like to discuss about one of the most heard names even in the morning, CRISPR-Cas9. It is the most effective, efficient, and accurate genome editing tool in all living cells utilized in multiple disciplines like medicine, agriculture, and biotechnology. Specifically, in agriculture, it could help in design of new grains to improve their nutritional value. It was first discovered in Japan in 1987, but not biologically functional at that time. At 2009, the CRISPR was experimentally conferred as adaptive immunity system in prokaryotes against viruses. Next slide, please. Conservation genomics has become a focal point for biodiversity research and has been increasingly recognized as a keystone towards the protection of biodiversity and natural resources for future generations of humankind. The deadliest combination of these approaches provide long-term survival of species, maintain genetic diversity, and provide a safeguard against extinction. Next slide, please. At this point, I would like to mention about genomic approaches in a plant conservation, a community that conserve plants, and much more activities are being performed by it. It is located, like this community is in Texas, US. Next slide, please. Next one. Yeah. By this, I conclude that tissue culture plays a vital role in genomic conservation by providing a controlled environment for propagation and preservation of plant genetic material, which enables efficient regeneration of plants, ultimately contributing for preservation of biodiversity. I would like to thank the head of the Department Agriculture and Rural Development, Dr. P. Lakshman Swami sir, our diverse faculty who are behind the successful writing of my article. And I would like to thank my team member, Mr. Sheila, who was effortfully making all these articles successful along with me. Thank you.
for a Sustainable Future LIFT 2025. This session marks the culmination of our collective efforts, knowledge sharing, and insightful discussions over the past 2 days. We are truly privileged to have with us the esteemed dignitaries, participants, and guests whose vitally important contributions have made this event a resounding success. As we draw this event to a close, let us take a moment to reflect on the insights and experiences we have gained, marking the crest of our collective journey. I now invite Nancy and group to lead us with a prayer song.
उसका गुणगान करो, इबादत करो, उसकी इबादत करो, इबादत करो, उसकी इबादत करो। दुनिया के लोगों ऊंची आवाज करो, गाओ खुशी के गीत, उसका गुणगान करो, इबादत करो, उसकी इबादत करो, इबादत करो, उसकी इबादत करो। याद रखोगे वही खुदा है, उनको ये जीवन उसने दिया है, उस जानकार से हम सब हैरान, हम जो सना के हम गीत गाए, रब का तुम शुक्र करो, ऊंची आवाज करो, गाओ खुशी के गीत, उसका गुणगान करो, इबादत करो, उसकी इबादत करो, इबादत करो, उसकी इबादत करो। यही सिलसिला है, उस पर ईमान धरो, उसके दर आप चलो, सखी इबादत करो इबादत करो सखी इबादत करो
Thank you, dear students. It is with great respect that I now invite convener of the conference, Dr. P.V. Rekha Devi, to deliver an overview of the proceedings over the past 2 days. We are eager to hear the reflections on the key discussions, insights, and achievements that have shaped this enriching event.
Good evening, everyone. It is my immense pleasure and privilege to read before you the overview report for the two-day international conference on innovations in life science research, harnessing transformative technologies for a sustainable future. The two-day international conference was successfully organized by the Department of Biotechnology over the past 2 days, that is 11th and 12th of March 2025. This event mainly aimed at bringing together the experts and leaders, researchers, students to discuss the transformative technologies in life science research. Day one started with an inaugural session, and it was convened by the presence of the chief guest, uh, Professor M.S. Reddy, and guest of honor, Dr. Georgie Reddy, Principal Loyola College, Kadapa. They delivered greetings and delivered a short message to all the participants, and that set a positive tone for the event. The keynote address was followed by this, and the keynote address was delivered by our chief guest, Dr. M.S. Reddy, on eco-friendly bio-inputs for secondary green revolution, which emphasized on sustainable practices in agriculture and their impact on the environment. Day one had three technical sessions by Dr. Maruti, the first technical session chair, who delivered a lecture on Innovate, Translate and Sustain the Future of Life Sciences, which focused on the integration of innovative solutions in life science research. The technical session 2 was chaired by Dr. Josna, who is a leading plastic surgeon and who delivered a talk on the biomaterials in the spotlight, pioneering sustainable practices in plastic surgery, which highlighted the advancements in biomaterials. The technical session 3 was chaired by Dr. S. Gajalakshmi, and she addressed on the microbial fuel cells for wastewater treatment, resource recovery, and energy generation. All the talks showcase the potential of the innovations in life science technologies for an environmentally sustainable research. The day concluded with 6 full-length paper presentations and 14 poster presentations by the students, which allowed the participants to share their research findings. Day 2 started with a theme that was focused on circular economic technologies, bioengineering, and biomanufacturing. The first session was chaired by Dr. Krishna Chaitanya Kari from Ethiopia, who discussed on the innovative approaches in chemistry for sustainable solutions, and he presented the novel methodologies in chemical research. Technical session 5 was chaired by Dr. J. Navina Lavanyarata, who was a principal in the degree and PG college at Nusibidu, and she delivered a talk on understanding fungal infections, their classification, diagnosis, and management. Technical session 6 was chaired by Dr. Kashi Vishwanath, who spoke on the application of nanotheranostics for advanced diagnosis and therapy of cancers, which emphasized the role of nanotechnology in cancer treatment. A special session on career guidance for studying abroad was conducted by I-20 Fever Consulting, which provided valuable information to the students and young researchers. The conference is now scheduled to be concluded with a valedictory session where we celebrate the success of exchange of knowledge and ideas for the past 2 days. We extend our heartfelt gratitude to all the speakers and participants for making successful and fruitful contributions to LIT 2025, and we look forward to seeing you for a continued impact of this conference on our global community, and we strive for sustainable solutions through innovative research. Thank you so much.
I now have the honor of inviting our esteemed principal, Dr. Sister Jacinta Quadras, to kindly grace the chair on the dais. Sister, please. We are now delighted to announce the winners of the oral and poster presentations. Is awarded to Miss D. Blessy from Central University of Andhra Pradesh, Anantapur.
What student? Here. Present outside.
Second, Miss Trisha Reddy from St. Francis College of Women, Hyderabad. I congratulate Miss Trisha Reddy for the second prize for oral presentation. We will send our prize to post. For the poster presentations, first prize is awarded to Miss M. Sucharashri from SRR and CVR College. Second prize for poster presentation goes to G. Kirti Priya, K. Pratima, and G. Swati from Agriculture and Rural Development, Stella College. We sincerely thank our esteemed principal, Dr. Sister Jacinta Quadras, for honoring us with your presence and for your unwavering support. Thank you so much, Sister. A short message from Sister.
I don't know, it's not a speech or a message. It is just I want to congratulate all of you students, especially those who have presented your poster presentations, and now some of you got prizes. I'm really proud of you that you took part in this international conference, and may this be a guide for you to climb higher in your career. Okay, and continue to do the research, innovate things. Okay, I wish you all the best, and I take this opportunity to congratulate Dr. Rekha and entire team, Hemashri, Gandaraju, Vaishnavi, Ponni, Supriya, and others also, all the other life science faculty. I think you, those who are present here, currently I'm not getting her name. Bharata Jyoti, okay, Bharata Jyoti, Krishna Manohar, and all the others who are here, any other faculty are here, Shravani, okay, and all the very best for you. Many more international conferences to come again. Continue to it. Maybe next chance is other life sciences you should take charge. Now this year it is taken care by biotechnology, who will be the next department? Students have to start. Initially, you should tell your teacher. Microbiology. Oh, let's see. But not in the this one, January, February, very busy months. You should start from July, August, September, within these 3 months. So now itself you have to plan, okay? So all the best. Wish you good time. I'm very proud of each one of you. All the best.
Thank you so much, sister. I now invite Dr. R. Gangaraju, Head Department of Microbiology, to deliver the vote of thanks.
Good evening, all. As we come to the conclusion of the International Conference on Innovations in Life Science Research Harnessing Transformative Technologies for the Sustainable Future, I have the honor of delivering the vote of thanks. First and foremost, I would like to express our heartfelt gratitude to our esteemed guests, keynote speakers, Father Dr. Jyoti Reddy and Professor M.S. Reddy. Their insightful contributions and inspiring words have set a remarkable tone for this conference and have ignited passion for innovation and sustainability among all participants. A sincere thanks to all the speakers who presented their valuable research for the past 2 days. Their dedication to advanced knowledge in life sciences is truly commendable. And the presentations have provided us with a wealth of information that will guide our future endeavors. I would also like to extend our appreciation to the organizing committee and volunteers who work tirelessly behind the scenes to ensure the smooth execution of this event. The commitment and hard work have made this conference a resounding success. All the participants, thank you for your active engagement and enthusiasm. Your questions and discussions have enriched our sessions and fostered a collaborative spirit that is essential for advancing research in our field. Lastly, I would like to acknowledge the support of management of Stella College, helping us in hosting this conference and providing us with such a conducive environment for learning and networking. I also thank and help rendered by the technical support, teaching and non-teaching staff, support staff, and IT personnel. As we conclude, let us carry forward the knowledge and connections we have gained here and continue striving towards innovative solutions for sustainable future. Thank you all once again, and we look forward to seeing you at future events. Thank you.
Thank you very much, sir. Now I kindly request all the dignitaries, participants, and attendees to rise for the national anthem.
गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Once again, thank you everyone.
అదే బ్యాగ్ మీద నేను ఎక్స్ప్లెయిన్ చేశాను శబరి ఈజ్ వెయిటింగ్ కోసం
ల్యాప్టాప్స్
ల్యాప్టాప్లో
ల్యాప్టాప్